Hey friends, welcome back. Pull your blanket a little closer tonight, settle in and get comfortable. Because what I'm about to share with you is the kind of story that will make you look at your own body, and maybe even your own family in a completely different way. Tonight we're going to talk about blood. Not in a scary way, in a fascinating, almost unbelievable way, because hidden inside the veins of roughly 1 in every 7 people on this planet is something scientists still cannot fully explain. It doesn't show up when you look in a mirror, but it has quietly shaped human history, triggered life and death moments in hospitals around the world, and sparked some of the most wildly debated theories in all of science. It is called Rh-negative blood, and here is the question that has haunted researchers for nearly a century. Every primate on Earth, every ape, every monkey, every chimpanzee, carries a specific protein on the surface of their red blood cells. Every single one. Except a small slice of humanity. Where did this difference come from? How did it survive tens of thousands of years when in certain pregnancies it could literally become a weapon? That a mother's own body turns against her unborn child? Why does it cluster so heavily in one ancient, isolated people in the mountains between Spain and France? A people whose language has no known relatives anywhere on Earth. Because by the time this story is over, you will know things about human blood that most people will never think to ask. Now, before we go any further, if this kind of deep, quiet storytelling is your kind of thing, if you love falling asleep to history that actually matters, I would love for you to hit that subscribe button right now and give this video a like. It costs you nothing. It means the world to me. And honestly, it's the reason I keep staying up late to put these stories together for you. If you feel moved to leave a comment down below, maybe tell me where in the world you're watching from, or what time of night it is for you right now. I read every single one, and there is something genuinely lovely about knowing you're out there. Now dim the lights a little if you can, let the room get quiet, and let's begin. New York City, 1937. Picture a cold autumn evening on the Upper East Side of Manhattan. The streets outside are alive with a kind of restless energy that only New York City can carry. Yellow taxis, the smell of roasting chestnuts from a corner cart, the deep rumble of the subway somewhere below the pavement. But inside a tall stone building on York Avenue, a laboratory is almost completely still. And in one of its upper floor rooms, a 69-year-old Austrian physician named Dr. Karl Landsteiner is bent over a laboratory bench, doing what he has done nearly every day for the past 4 decades. Now, if you have never heard of Karl Landsteiner, let me take just a moment to introduce you, because this man is one of the most important scientists who ever lived, and almost nobody outside of medical school knows his name. Back in the year 1901, when Landsteiner was still a young doctor working in Vienna, he did something that changed medicine forever. Before that moment, doctors had been trying to transfuse blood from one person to another for centuries. Sometimes it worked, and sometimes the patient died, screaming their body going into violent shock. He identified the first 3 human blood types, which he called A, B, and C, though we now call that third one O, and he showed that when you mix incompatible types together, the red blood cells clump and destroy each other. It was a discovery so profound that it eventually earned him the Nobel Prize in Medicine in 1930. By the time we find him in that New York laboratory in 1937, Landsteiner is officially retired. His wife Leopoldine had been diagnosed with thyroid cancer, and some who knew him believed he threw himself even harder into his research, the way certain people do when the rest of life becomes too painful to sit still with. Research the way certain people do when the rest of life becomes too painful to sit still with. And in the autumn of 1937, he and his young colleague, Dr. Alexander Wiener, were doing something that seemed on the surface fairly routine. They were injecting rabbits and guinea pigs with the red blood cells of rhesus macaque monkeys. You've probably seen them in documentaries, curious little creatures that often live near temples in India, stealing bananas from tourists with absolutely no shame. When you inject animal blood into a rabbit's body, the rabbit's immune system reacts and creates antibodies, protein weapons designed to fight off what it sees as a foreign invader. Those antibodies can then be collected from the rabbit's blood and used as a tool, a detector really. You take those rabbit antibodies and mix them with a human blood sample, and you watch to see what happens. If the blood clumps and reacts, it means the human blood contains an antigen that matches what was in the monkey. If nothing happens, the blood is clear. What Landsteiner and Wiener found when they began testing human blood samples was deeply strange. About 85% of the people they tested reacted. Their blood clumped. Their blood contained the same surface protein that the rhesus monkey's blood carried. But here is the part that made both men go very quiet. The remaining 15% of people they tested did not react at all. Their blood stayed perfectly smooth. No clumping, no reaction, nothing. It was as if for those 15 people out of every 100, the rhesus monkey test simply did not apply to them. Their blood was missing something that virtually every other primate on Earth carries as a matter of biological routine. Landsteiner stared at those clear, unclumped samples and wrote in his notes that they merited further investigation. That is scientist language, for this is very, very strange, and I am not sure what to do with it yet. Now I want to pause here for just a second, because I think it is worth really sitting with what that discovery meant. Imagine you are building a family tree of all living creatures. You go back far enough, hundreds of millions of years, and every primate on Earth shares a common ancestor. Monkeys, apes, chimpanzees, humans. We're all cousins in the long, slow language of evolution. And through that shared ancestry, almost every branch of the primate family tree carries this particular proteins, this Reich factor on the surface of their red blood cells. It is ancient, it is deeply conserved. It is biologically speaking something that has been passed down across unimaginable stretches of time. And yet, 1 in every 7 human beings does not have it. Not one other primate species shows this absence. Just us. Just a small specific slice of humanity. If blood tells the story of where we come from, then Rh negative blood is a chapter that does not seem to belong in the same book as everything else. Now Landsteiner and Wiener published their findings in 1940 in the Proceedings of the Society for Experimental Biology and Medicine. It was a short paper, just 2 pages, and the scientific world, which was by then consumed with the growing shadow of World War II, barely noticed. But the implications of those 2 quiet pages would take decades to fully unravel, because almost immediately doctors began to realize that the Rh factor was not just a biological curiosity. It had real, urgent, sometimes devastating consequences. The first clue came from blood transfusions. Some patients who received blood that was supposedly the right type perfectly matched for ABO compatibility still went into transfusion reactions. If a patient was Rh negative and received Rh positive blood even once, their immune system would begin quietly building antibodies against it. The first transfusion might seem fine, but the body was making a note, cataloguing the threat, preparing. And if that patient received Rh positive blood a second time, their immune system would launch a full attack. The donated red blood cells would be destroyed. The patient would go into shock. In the worst cases, they would die. It was in a strange way a kind of immunological memory that had turned against its own host. But the deepest consequence of the Rh discovery was not about transfusions at all. It was about mothers and their unborn children. And this part of the story I want you to really hear, because for hundreds and hundreds of years, quietly and visibly, without anyone understanding why, a particular kind of tragedy had been unfolding in families around the world. A woman would have her first baby, everything would seem fine. The baby would be born healthy and alive, and the family would celebrate. But then with the second or third pregnancy, something would go terribly wrong. Doctors would note the jaundice creeping across the newborn's skin within hours of birth. They would note the unusual fragility of the infant's blood. They would try transfusions, try everything they knew. And in many cases they would lose the baby anyway. The condition even had a name by 1932. A doctor named Louis Diamond gave it one erythroblastosis fetalis. The red blood cells of the fetus were being attacked and destroyed, forcing the baby's body to pump out immature, abnormal blood cells in a desperate attempt to keep up. A French midwife had first written about a similar pattern as far back as 1609. A set of twins, one born pale and swollen, the other fine, both from the same mother. 3 centuries of mystery, 3 centuries of grief, 3 centuries of babies dying without anyone understanding why. That means more than half the babies born with this condition in the 1930s did not survive their first few days of life. And the mothers who lost them, imagine what it must have been like to have a healthy firstborn and then watch, helplessly as each subsequent pregnancy ended in heartbreak, with doctors shaking their heads and offering no explanation. That kind of loss leaves marks on families that last for generations. The answer, when it finally came, arrived through the work of Dr. Philip Levine. In 1939, 1 year before Landsteiner and Wiener's paper was published, Levine examined a specific haunting case. To help her recover, doctors gave her a blood transfusion, and because her husband was present and willing, they used his blood. Same ABO type, should have been safe, but she reacted violently to her own husband's blood. He suspected it was related to the pregnancy, that the woman had somehow become sensitized to an antigen from the fetus's blood which had crossed the placenta and entered her circulation. That antigen, he would later confirm, was the Rh factor. Here is what was happening. When an Rh-negative mother carries an Rh-positive baby, a baby who has inherited the Rh factor from its father, tiny amounts of the baby's blood can cross the placenta, especially during delivery. The mother's immune system, encountering this foreign protein for the first time, begins producing antibodies against it. The first pregnancy often ends fine because the immune response is slow and the baby is born before enough. Antibodies build up to cause serious harm. But in the next Rh-positive pregnancy, those antibodies are already loaded waiting. They cross back through the placenta, a mother's own immune system doing exactly what it was designed to do, protect her body from foreign invaders turning against her own child. It is one of the most quietly devastating ironies in all of human biology. Now here is where I want to take a small, gentle step back and ask you the question that is at the heart of everything we will explore together in this series. Because every scientist and every armchair philosopher who has ever spent serious time thinking about Rh-negative blood eventually arrives at the same uncomfortable puzzle. If carrying Rh-negative blood can cause this kind of immunological conflict during pregnancy, if it can in certain circumstances result in the death of a child, then why hasn't natural selection eliminated this trait? Over thousands of generations, traits that reduce reproductive success, traits that kill offspring, tend to disappear from a population over time. Nature does not keep mistakes around. And yet here we are, thousands of years into recorded human history, and Rh-negative blood is still here, still present in roughly 15% of the global population. The mainstream scientific answer, that it is simply a random genetic mutation that has persisted due to the complexity of natural selection, is technically plausible. Because when you start to look at where Rh-negative blood actually lives in the world, where it clusters, where it concentrates, which populations carry it at the highest rates, a picture begins to emerge that is genuinely difficult to ignore. And that picture leads us somewhere very old, somewhere very isolated, to a mountain range between two countries, to a people whose origins no linguist or geneticist has been able to fully explain, to the Basque. But that is a story for the next part of our journey. For now, I want to leave you here in the quiet of Dr. Landsteiner's laboratory, with 2 glass slides, 2 blood samples sitting side by side, one that reacted to the rhesus antigen, and one that did not. One that belongs to the long unbroken chain of primate biology stretching back millions of years, and one that simply does not. Carl Landsteiner died 6 years after his discovery. On the 26th of June 1943, he had a heart attack in his laboratory. He died with a pipette still in his hand, working until the very last moment, still trying to understand the blood. I think about that image sometimes. An old man in a quiet room holding a glass tube full of questions he never got to answer. Some of those questions we are still asking today. Stay with me. The next part of this story goes even deeper, and what waits in those mountains is unlike anything you have heard before. Somewhere in Western Europe, wedged between two countries that have spent centuries trying to define their borders, there is a place that has quietly refused to be defined at all. No empire ever fully absorbed it. No wave of migration ever fully replaced the people living there. No linguist in over 200 years of trying has ever been able to trace the roots of the language spoken within it to any other language on earth, and the blood flowing through the veins of its people, that too belongs to a category all its own. We are talking about the Basque country, and if you have never spent much time thinking about the Basques, I want to spend some time with you here, because of all the threads that run through this mystery. This one is the one that made me stop whatever I was doing and just sit with it for a while. Let me paint you a picture first. Imagine the western end of the Pyrenees mountain range, that dramatic spine of rock that separates Spain from France along the top of the Iberian Peninsula. Right there, where the mountains begin to lower themselves toward the Bay of Biscay, where green hillsides roll down to a coastline that smells of salt and ancient fishing boats. That is where you find them, the Basque people. About 3 million of them today, scattered across what is now the northern corner of Spain and the southwestern edge of France. They have their own food traditions, their own festivals, their own flag, their own fierce and ancient pride. But what sets them apart from every other group of people in Western Europe, what has puzzled scholars for generations, is something you cannot see when you look at them. It is their language. It is called Euskara. And I want you to understand just how strange a thing this language is. Every major language spoken in Europe today belongs to a single, enormous family of related tongues known as the Indo-European languages. English, Spanish, French, German, Russian, Greek, Italian, Hindi, all of them, despite sounding so different from one another, are cousins. They share common ancestors. Shared roots for basic words, words for mother, for water, for fire. A shared grammatical logic buried somewhere beneath centuries of change. Linguists can trace the family tree. They can point to a proto-language spoken by people on the Eurasian steppes, somewhere around 6,000 years ago, that eventually spread and evolved into almost every tongue in Europe and large parts of Asia. Now take Euskara. Pick up any Basque word at random, the word for house etxie, the word for man gizona, the word for summer uda. Now try to find those words anywhere else. In Latin, in Greek, in Germanic, in Celtic, in Slavic. You will find nothing, not even a distant echo. Linguists have a precise technical term for this situation. They call Euskara a language isolate, meaning it has no known living relatives, no known extinct relatives either, no demonstrable connection to any other language family anywhere on Earth. And this is not a small puzzle. This is a question that has consumed entire careers. The Basque language, according to all available evidence, predates the arrival of every other major linguistic tradition in Western Europe. At minimum, it is older than 12,000 years, since it clearly predates the spread of Indo-European languages. Some researchers believe it is far older than that. Some believe it may carry the echo of words spoken by people who painted the walls of limestone caves during the last ice age. Think about that. A language potentially old enough to have been spoken by people who watched woolly mammoths cross the valleys below those Pyrenean peaks. And it is still alive. Roughly 850,000 people speak it today. Now here is where the blood comes back into the picture. Because when researchers in the mid-20th century began systematically mapping the distribution of Rh-negative blood across Europe, going region by region, population by population, they found something they were not expecting. the Basque country was off the chart. Globally, H-negative blood appears in roughly 15% of the population. In most European countries, the number is somewhat higher. The United Kingdom sits around 17%, Germany, a little lower France, somewhere in between. But the Basques, between 30 and 35% of the Basque population, carries R-negative blood, more than double the global average. And in some of the most remote, isolated Basque villages, the places where the language is best preserved, where outside contact has historically been least frequent, the rates have been recorded even higher. To put that in perspective, if you walked into a random cafe in Paris or Madrid and tested everyone's blood, roughly 1 in 6 or 7 people would be Rh-negative. Walk into a cafe in a traditional Basque village, you're looking at 1 in 3, sometimes more. Now, does this mean the Basques are strange or special in some mystical sense? I want to be careful here because this is where the internet tends to go off the rails pretty quickly. Type Basque blood into a search engine, and within about 30 seconds you will find claims ranging from the Basques are descendants of Atlantis, to Rh-negative blood is proof of alien ancestry, to my personal favourite Basques are the original humans, and everyone else is a later genetic modification. I'm not saying those things, and with respect, neither should you. What I am saying is something far more interesting. I am saying that when you find a population with a specific genetically unusual trait occurring at more than twice the global rate, and when that same population also speaks a language with no known relatives, and when both of those anomalies have persisted for what appears to be an extraordinarily long time, then you have to ask what preserved them, and the answer that science is increasingly pointing toward takes us back to something very cold, very dark, and very, very old. 35,000 years ago, the lands that are now the Basque country were home to Cro-Magnon people, not Neanderthals. Neanderthals were already disappearing by this point, their populations contracting, their world slowly shrinking. Cro-Magnon is our own species, Homo sapiens sapiens, anatomically modern humans, us essentially standing in furs beside a river that no longer has the same name. And these particular people, the ones who settled the Franco-Cantabrian region, that stretch of land running from northern Spain up through what is now southwestern France, were remarkable. They were the painters, the Lascaux cave paintings in southern France, the extraordinary art at Altamira in northern Spain just west of the Basque country, hundreds of other decorated caves spread across the same region. These were their hands, their minds, their sense of beauty and meaning pressed into limestone walls in the flickering orange light of fat-fueled torches. Some of this art is 35,000 years old, some of it is older, and here is something worth sitting with. For much of that time, the same region served as one of the primary refuges for human populations during the last glacial maximum, the period roughly 20,000 years ago, when ice sheets pushed down from the north and most of Europe became effectively uninhabitable. The Franco-Cantabrian zone, sheltered by mountains to the north and warmed by the Atlantic current, remained livable. People stayed. While others fled or died, populations clustered in this corner of the continent survived. Geneticists today refer to this as a glacial refugium, a refuge in ice. A place where ancient genes, including researchers, now believe the earliest Rh-negative alleles in Europe were preserved in isolation, while most of the surrounding human world was being reshuffled. When the ice finally retreated around 12,000 years ago, something happened that completely transformed the genetic landscape of Europe. New people arrived, Neolithic farmers spreading northward and westward from the Near East, carried a different set of genes, a different set of blood types, different agricultural tools and different ideas. They mixed with the existing hunter-gatherer populations across most of Europe, In most places, the incoming farmer genetics eventually became dominant. The older hunter-gatherer blood, the older languages, the older ways were diluted, absorbed, overwritten. But in that western corner behind the natural fortress of the Pyrenees, something different happened. The mixing was far less complete. A 2021 study led by researchers at Pompeu Fabra University and the Institute of Evolutionary Biology in Spain analysed over 600,000 genetic markers across the Basque genome, one of the most comprehensive studies of Basque genetics ever conducted. Their conclusion: the Basques' genetic uniqueness is not because they came from somewhere different. It is because they stayed where they were. While the rest of Europe was being layered, farmers over hunter-gatherers, later migrations over earlier ones, empire over tribe, the Basque country remained in genetic and cultural terms relatively continuous. Their mountains kept them, their language kept them, and perhaps in a quietly circular way being kept also preserved the blood. Pause here for a moment because I want to ask you something. Have you ever thought about what it would feel like to carry a language inside you that no one else on earth truly shares? I mean that in the deepest sense, not a rare language that happens to have few speakers, but a language with no living relatives at all, no cousins, no distant aunts, no linguistic DNA in common with anything else. When a Basque person speaks Euskara, they are not reaching across a bridge to any other tradition. They are simply standing alone in a very old room. And I think that image matters when we think about the blood, because both things, the language and the blood, point toward the same conclusion. This is a population that was for a very long time separated from the major currents of human mixing. Not by choice necessarily, by geography, by mountains and sea and the particular stubbornness of people who have always known exactly who they are. There is another detail that keeps coming up in research on this topic, and it is one that I find genuinely striking. Other places in the world with elevated rates of Rh-negative blood share something with the Basque country. The Atlas Mountains of Morocco, where the Berber populations have historically been concentrated, show Rh-negative rates between 15 and 20%, notably higher than the surrounding regions. The remote Scottish Highlands, the Western Islands of Ireland, parts of Sardinia, the Canary Islands off the coast of northwest Africa. What do many of these places have in common? They are refuges, mountain ranges, islands, coastal extremities, places where historically people got left behind by the great movements of migration and empire, places where the deep old genetic layers were not fully covered over. Researchers have begun to refer to this pattern using the idea of Western hunter-gatherer ancestry, the genetic signature of the people who lived in Europe before agriculture arrived, and before the later steppe migrations reshuffled things further. A 2024 genomic study found that Western hunter-gatherer ancestry, which is present in higher concentrations in the Iberian refugium that includes the Basque country, appears to correlate with specific longevity markers in modern populations, which is, if you think about it, its own kind of mystery. Blood that is hard to explain, in people who have always been hard to explain, lasting longer than anyone had any right to expect. I want to come back to that word survive, because the Basque people as a culture have done something that seems almost impossible from a historical standpoint. They have survived the Roman Empire. Caesar's legions moved through the Iberian Peninsula. They conquered almost everyone. The Basques. The Romans left them largely alone. Not because the Romans were kind, but because the mountains were steep, and the people were fierce, and the cost of full conquest outweighed whatever the Romans thought they might gain. They survived the Moorish occupation of Iberia in the 8th century, which again swept across most of the peninsula, reshaping language and culture across Spain and Portugal. The Basque country remained. They survived centuries of Spanish and French crown pressure. They survive Francisco Franco, who banned the Basque language during his dictatorship in the mid-20th century, actually made it illegal to teach it, speak it in public, or use it in official settings. He thought he could erase them. He failed. By 1975, the year Franco died, Euskara had survived. Battered, reduced, but alive. And in the decades since, it has made a remarkable recovery. Today it is co-official alongside Spanish in the Basque country, taught in schools, spoken by around 1 million people, and embraced by a new generation with a ferocity that would have surprised even its oldest defenders. A language that should have died several times over. Still here, much like the blood. Now I want to be honest with you about what science does and does not currently tell us. The honest answer is this: scientists understand the Rh-negative trait at the genetic level reasonably well. It results from a specific deletion in the gene responsible for producing the Rh factor protein, a deletion that happens to be far more common in Western European populations and extraordinarily common in the Basques. But why that deletion became so concentrated in this particular population, why it hasn't been slowly diluted out over thousands of years, the way most recessive traits tend to be, is something researchers are still actively working on. The most rigorous scientific explanation currently centers on isolation. If a small population carries a trait at higher than average frequency and then becomes genetically isolated, that trait can become even more prevalent through a process called genetic drift, essentially random fluctuation in a small gene pool with a bit of chance deciding which traits stick around in high numbers, combined with the possibility that there may be some advantage to the Rh-negative trait in certain environments, perhaps related to resistance to specific diseases or immune function differences not yet fully characterized. You have a plausible framework for how this could happen. But plausible is not the same as fully understood, and there are elements of the Basque anomaly that the isolation plus drift explanation does not neatly resolve. The extreme concentration of Rh-negative blood specifically in this region, specifically in this population, specifically correlated with the linguistic uniqueness found nowhere else on Earth. That combination still raises questions that honest researchers acknowledge they cannot fully answer. Here is one more thing worth knowing before we move forward. The Basques themselves have a creation story. In many versions of it, the original Basque person, the mythological ancestor, is simply said to have appeared in the Pyrenees, not descended from other peoples, not arrived from elsewhere, simply there. From the beginning. Now I am not telling you that myth is biology. Myths are not genetic studies and creation stories are not historical records, but there is something quietly telling about a people whose oldest stories say we were always here. We came from no one. We belong to this place the way the stone belongs to the mountain, and when you look at what modern genetics and blood research actually shows, that yes, in fact, this population carries genetic material that traces back to some of the oldest continuous human habitation in Europe. The myth and the science are in their own way pointing in the same direction. Some things are older than anyone thought to ask about. We have been following a thread tonight that began in a New York laboratory with a retired scientist who refused to stop working. And now it has brought us to a mountain range on the edge of the Atlantic, to a people with blood that doesn't fit the map, and a language that belongs to no family on Earth. But here is the thing about mysteries. The deeper you go, the more questions you find. Because the Basques are only one piece of this. Their negative trait does not only live in the mountains of the Pyrenees, it lives in the caves behind those mountains. It lives in bone and ash and ancient sediment in the genetic record of people who painted pictures of bison and aurochs on stone walls by firelight 35,000 years ago. And what those ancient people tell us about how this blood began, about where the very first Rh-negative humans lived, what world they moved through, And what forces may have driven this trait forward across the centuries, that story is waiting for us, just ahead. Close your eyes for a moment. Actually, maybe don't close your eyes, because then you won't be able to keep listening. But bear with me. Picture complete darkness. Not the darkness of a bedroom with the curtains drawn. Not the darkness of a power outage in a city where there is still the faint glow of streetlights under the door. I mean absolute darkness. The kind that presses in on you from all sides and makes you feel as though the space itself has weight. Now add a single flame, a small fire burning inside a hollowed stone fueled by animal fat throwing a warm and flickering circle of orange light onto a wall of limestone, and there crouching with a piece of ochre or charcoal in hand a human being, deliberate, patient, breathing slowly, working. This is the world we're stepping into now, not the world of laboratories and Nobel Prizes, not the mountain villages of the Pyrenees with their ancient language and their fierce political identity. We're going much further back than that, 35,000 years, give or take, to the people who were here first, the ones who in a very real genetic sense may have been carrying the blueprint of Rh-negative blood before the word blood existed in any language. Their name Cro-Magnon comes from a French rock shelter called Abri de Cro-Magnon in the Dordogne region of southwestern France, where their bones were first formally identified in 1868. But calling them by that name sometimes gives people the wrong impression. It makes them sound primitive, distant, barely recognizable as human. They were not. Cro-Magnon people were anatomically identical to you and me. Same brain size, same vocal apparatus, same hands. Tall often, some skeletal remains show individuals over 6 feet in height, which would have been remarkable for any era. They buried their dead with ceremony. They used needles made from bone to sew fitted clothing. They made musical instruments. They planned hunts that required coordination across large groups over long distances. And they made art. Art of a quality and ambition that genuinely staggers the imagination when you stand in front of it. September 12th, 1940. Nazi Germany had invaded France just months earlier. In the small village of Montignac in the Dordogne, a 17-year-old boy named Marcel Ravidaille was walking in the woods near a hill called Lascaux when his dog, a terrier named Robot, which is just a wonderful name for a dog, disappeared into a hole left by a fallen tree. Marcel went after him. He squeezed through the gap, dropped down a 50-foot shaft, and found himself standing inside a cathedral. That is not a metaphor. The walls and ceilings of the Lascaux cave complex were absolutely covered in paintings. 600 paintings, nearly 1,500 engravings. Horses, deer, bison, ibex, wild cattle, lions, a rhinoceros, a bear. Enormous, confident, beautifully observed animals rendered in ochre, red and yellow and black and brown, filling the vaulted stone ceiling with a kind of energy that people who have stood in the original cave describe as overwhelming. One gallery, the Hall of the Bulls, contains 4 massive aurochs, the largest prehistoric cave paintings ever found, each one several meters long, flowing across the rock as if in motion, as if the stone itself is alive. The paintings at Lascaux date to approximately 17,000 years ago, but the art tradition that produced them was already ancient by then. The cave paintings at Altamira in northern Spain, just west of the Basque country, a detail I want you to hold on to, date back around 36,000 years. The Chauvet Cave in southern France, discovered in 1994, contains art that is even older. Some images there push past 40,000 years. 40,000 years! To put that number in perspective, all of recorded human civilization from the earliest Sumerian writing to the smartphone in your pocket fits inside roughly 5,000 years. These paintings predate that by 8 times over, and they were made by people who were, as far as we can determine, fully human in every sense that matters. Now here is the question that has been gnawing at researchers for decades. Who exactly were these people? Where did they come from? What happened to them? And most critically for our purposes tonight, what does their ancient blood tell us? For most of the 20th century, the standard answer was fairly simple. Cro-Magnons came into Europe from somewhere to the east out of the Middle East, broadly speaking around 40 to 45,000 years ago. They spread westward and northward across the continent. They displaced or absorbed the Neanderthals who had been there before them. They thrived. And then roughly 10 to 12,000 years ago, when Neolithic farmers began arriving in Europe from Anatolia, modern Turkey, those early hunter-gatherers were themselves largely replaced. End of story. Except that is not quite what the genetic evidence shows. Not in this corner of Europe. In 2023, a team of paleogeneticists published a landmark study in the journal Nature, the largest ancient DNA analysis of Ice Age Europeans ever attempted at that point. And what they found specifically in the southwestern corner of Europe, the Iberian Peninsula, the Franco-Cantabrian zone, the exact region where those extraordinary cave paintings were made, told a different story from the rest of the continent. In most of Europe, the ancient populations who had survived the last glacial maximum were indeed largely replaced by incoming Neolithic farmers, and later by Bronze Age steppe migrants. The genetic landscape was overwritten again and again. But southwestern Europe, the glacial refugium, the shelter behind the Pyrenees, retained a thread of continuity that was measurably different from everywhere else. A 23,000-year-old individual whose remains were found in southern Spain at a site called Cueva del Malalmuerzo carried genetic ancestry that connected Aurignacian people from before the last glacial maximum with the western hunter-gatherers who came after it. Genetic continuity preserved in the ice and the mountains spanning tens of thousands of years, and that same western hunter-gatherer ancestry, when ancient blood groups were finally reconstructed from ancient DNA in 2017 by a research team analyzing prehistoric European genomes, showed something striking. Mesolithic hunter-gatherers, the descendants of those Ice Age people, had reached negative allele frequencies higher than any population alive today. Let that settle in for a second. The people who made the cave paintings, the people who sheltered in that southwestern corner of Europe while the glaciers carved the north, the people who in the Iberian Peninsula and the Franco-Cantabrian zone show remarkable genetic continuity across 20,000 years of recorded human history. Those people carried Rh-negative blood at rates that modern populations, even the Basques, do not reach. Now I want to be honest about what this means and what it does not mean. It does not mean that Rh-negative blood is inherently ancient and Rh-positive blood is somehow newer or less authentic. Both are part of the human story. What it does mean is that the distribution of Rh-negative blood in modern populations appears to reflect the degree to which people have retained ancestry from those oldest Western European hunter-gatherers, in populations that mixed heavily with incoming Neolithic farmers who carried very low rates of Rh-negative blood closer to 4%. The overall frequency dropped toward the global average. In populations that for geographic or cultural reasons mixed less thoroughly, the Basques, certain Scottish Highland communities, the Berber populations in the Atlas Mountains of North Africa, the older frequency held on. It is in a very literal sense a bloodline running backward through time, a thread connecting modern people to those flickering firelit rooms in southern France and northern Spain. So who are these Ice Age people really? What was daily life like for the individuals who painted those walls? This is the part of the story that gets genuinely intimate, and I find it one of the most moving things to think about. They lived in small bands. Probably between 20 and 50 people in a group, though bands almost certainly came together seasonally in larger gatherings. There is archaeological evidence for communal sites, where many different groups met, traded, exchanged mates, and shared knowledge. They were not wandering randomly. Their movements were strategic and seasonal, following the migrations of the herds they depended on. Red deer in summer moving to higher ground, reindeer and wild horses in autumn, back to sheltered valleys in winter where caves provided protection from temperatures that could drop dramatically in Ice Age Europe. They were intensely observant of the natural world in ways that we living in climate-controlled spaces and getting our food from refrigerators can barely begin to imagine. The accuracy with which the Altamira ceiling paintings capture the physiology and behavior of bison, the way a bison tucks its legs when it rolls on the ground, the precise arc of a horse's neck in motion, suggests people who had spent their entire lives watching these animals with the focused attention of those who had no other choice. Their survival depended on understanding. Their art was an extension of that understanding. And this is where I want to offer you something that is my own perspective, not a scientific fact, just a thought I keep coming back to when I think about this subject. There is a tendency, even among well-meaning people, to think of prehistoric humans as incomplete, as if they were drafts of a person rather than the full thing, as if the refinement of civilization was something being gradually added to a rough early model. But when you stand in front of a 20,000-year-old horse painted on a cave wall, and the proportions are correct and the line has confidence and grace, and whoever made it clearly understood something deep about the animal they were drawing, you stop thinking of the person who made it as incomplete. They were fully here, fully conscious, fully complex, and the fact that some thread of what flowed through their veins may still flow through the veins of people alive today, including possibly people watching this very video, is not a comforting piece of mythology. It is a documented genetic reality. Let me give you one more specific piece of this picture because I think it matters. The cave at Altamira sits in the Cantabrian region of northern Spain. The Basque country begins roughly 80 kilometers to the east. This is the same cultural zone, the same geographical pocket, the same people moving across the same landscape over the same tens of thousands of years. When archaeologists study the succession of cultures across the Franco-Cantabrian region, the Aurignacian culture, then the Gravettian, then the Solitrian, then the Magdalenian, they find remarkably consistent stylistic and technological continuity. These were not disconnected waves of people replacing each other. They were descendants inheriting from their predecessors, building on what came before. The Magdalenian culture, which produced some of the most sophisticated cave art of all, including the masterworks at Lascaux, appears to have developed locally in this very region, without significant outside influence. In genetic terms, the people of the Franco-Cantabrian zone during the Upper Paleolithic represent one of the most continuous human populations in European prehistory. A gene pool that stayed relatively intact across an almost incomprehensible span of time, and within that gene pool preserved in limestone caves and ancient sediment, and the yellowed calcite of old bones, ancient Rh, negative alleles waiting to be rediscovered. Not by ancient astronaut theorists, not by fringe genealogy websites, but by paleogeneticists extracting DNA from prehistoric skeletal material, running it through sequences, and finding in the data what the data quietly, patiently insists is there. You might be wondering, and I think this is a fair question to ask, what exactly is it that makes the Rh negative trait persist? If it caused problems in pregnancy as we explored earlier, why didn't it simply fade away over those tens of thousands of years? The full answer to that question is still unresolved, and we're going to spend real time on it later in this journey. But here is one piece of the puzzle that is worth planting now. For most of human prehistory, second and subsequent pregnancies that ended badly, the erythroblastosis fetalis that we talked about, would not necessarily have been understood as connected to the same mother's previous pregnancy. Infant loss was common enough that its specific cause was rarely investigated. The genetic penalty for carrying Rh-negative blood was real but diffuse, spread across generations, diluted by the general harshness of prehistoric life. And against that diffuse penalty, researchers are now beginning to believe there may have been compensating advantages that we are only beginning to understand. The ancient DNA showing that hunter-gatherer populations carried Rh-negative alleles at higher rates than modern populations also shows something else. Those populations were not dying out. They were surviving in some of the harshest conditions Europe has ever seen. Surviving the last glacial maximum, surviving the Neolithic transition, surviving long enough to leave their genetic mark on modern people scattered from Ireland to Morocco to the Scottish Islands. Whether the Rh-negative trait itself was advantageous in some way, or whether it simply travelled alongside other traits that were, is a question scientists are actively working on. But the survival of the people who carried it is not in question. They survived. The blood survived with them. There is one last image I want to leave you with from this part of the story. Inside the Lascaux cave, among the hundreds of paintings of animals, there is exactly one human figure. Just one. He is roughly drawn compared to the spectacular animals surrounding him, almost hastily sketched as if the artist spent all their care on the bison and the horses, and saved only a quick mark for the human presence. He appears to be falling, or perhaps in the act of reaching for something just beyond his grasp. Archaeologists have debated his meaning for 80 years without reaching consensus. Is he a shaman, a hunter, a figure from a story that no one alive can any longer read? Nobody knows. But here is what I keep coming back to. Whoever painted him standing in that cave with a lamp made of animal fat reaching up to press pigment into limestone was as fully human as you are, as curious, as creative, as aware of their own smallness in the face of something vast and ancient and only partially understood. And something of what they were, something carried in the deep molecular structure of what made them who they were, may still be here, in veins that have never been told where they came from. We have come a long way from a New York laboratory, from a mountain range in the Pyrenees, from ice and firelight and painted bison on limestone walls, and we've barely scratched the surface. Because everything we have explored so far, the science, the geography, the ancient blood, eventually leads to a question that no scientist has been able to escape. If this trait has persisted across 40,000 years, written into the most ancient layers of human DNA, why? What kept it alive? Was it random chance, as many researchers insist, or was something else happening? Something happening in the very mechanism of human reproduction, in the space between mother and child, in the biology of survival itself. That question sits at the heart of what comes next, and it is far more personal than anything we have talked about so far. There is a phrase that has echoed through palaces and throne rooms for centuries: royal blood. Kings used it to justify their power. Courts used it to police who could marry whom. Armies were raised in its name. Wars were fought over whose bloodline had the stronger claim to a crown. But here is the thing nobody standing in those gilded halls seemed to notice, or perhaps nobody dared to say out loud. The blood of Europe's royal families was in very measurable, very biological ways quietly destroying itself. Not metaphorically, not symbolically, in the literal cellular, laboratory testable sense. The obsession with keeping royal blood pure, untouched by commoners, undiluted by outsiders, circulating only within an ever-tightening circle of aristocratic marriage, had over generations turned the act of inheritance into something almost indistinguishable from a slow genetic unravelling. And the story of how that happened is one of the most extraordinary, darkest, and if you will forgive me for saying this, occasionally most darkly comedic chapters in all of European history. Let's start at the top, or rather at the jaw. If you have ever seen a portrait of King Charles II of Spain painted in the late 17th century, you will understand immediately what I'm about to describe. He had a jaw that did not belong on a human face. Not in a cartoonish way. Not in the way a child might exaggerate a drawing, but in a way that made eating genuinely difficult, that made speaking nearly impossible to understand, and that made him the most physically arresting, and in some rooms the most alarming person anyone in his court had ever seen. His lower jaw protruded so severely that his teeth could not meet. His tongue was so large it caused him constant trouble with basic speech. A British envoy named Alexander Stanhope, who visited him in the 1690s, wrote that the king had what he described as a ravenous stomach, and noted that he swallowed everything whole, because his upper and lower teeth could never actually make contact. Stanhope was being diplomatic. The medical reality of Charles II's condition was considerably more harrowing than polite diplomatic correspondence was willing to acknowledge. His autopsy, conducted after his death in the year 1700 at the age of 38, reportedly found a heart described as being the size of a peppercorn. His lungs were corroded. His intestines were gangrenous. One testicle had reportedly atrophied to a state his physicians could barely describe in professional language. He had no children. He never had children. The entire Spanish Habsburg dynasty ended with him. His nickname given to him by his own people was El Hechizado, the Hexed. Now why am I telling you this? Because Charles II of Spain was not born into misfortune by accident. He was the product of something that can now be measured. A team of researchers from the University of Santiago de Compostela in Spain spent years analysing family trees spanning 16 generations and more than 3,000 individuals connected to the Habsburg dynasty. They calculated an inbreeding coefficient for each member of the family. For context, the inbreeding coefficient for two first cousins having a child together would be about 0.06. For Charles II of Spain, it was 0.25, more than 4 times that. His parents were a niece and her uncle. His mother was also in a different branch of the family tree, his first cousin. If you tried to draw his family tree in a clean branching diagram, it would not branch at all. It would loop back on itself so many times the diagram would barely be readable. 9 of the 11 marriages made by the Spanish Habsburgs over 184 years of rule involved close relatives. 9 out of 11. The result was not one failing dynasty. It was a dynasty that systematically concentrated every harmful recessive trait it carried into a single increasingly fragile bloodline generation by generation, until the final branch simply stopped. Historians and geneticists now broadly agree that the downfall of the Spanish Habsburg dynasty was not caused by enemies, by bad politics, or by divine punishment. It was caused by biology, by the thing they were most trying to protect. But here is where I want to slow down and make a distinction that I think matters enormously. The story of the Habsburgs is not strictly speaking about Rh-negative blood. The Habsburgs were not undone by blood-type incompatibility. They were undone by inbreeding, by an entirely different mechanism. So why are we here? Because what the Habsburg story illustrates better than almost any other case in history is the deep, strange, and often dangerous power that blood genetics can hold over human lives, human dynasties, and even human civilizations, and it opens a door to something that is directly connected to our main thread, because while the Habsburgs were busy destroying themselves in Madrid, a very different royal family roughly 1,100 kilometers to the northwest was doing something that would eventually alter the fate of the Russian Empire, and that story involves blood in the most literal possible way. Queen Victoria of Great Britain was born in 1819. She became queen at the age of 18. She reigned for 63 years, the longest reign of any British monarch up to that point, and she outlived a husband, several of her own children, and most of her contemporaries. She was by any measure one of the most powerful people on the planet during the second half of the 19th century. She was also a carrier of haemophilia. Now, a carrier is not the same as someone who has the disease. Haemophilia, a condition in which the blood lacks certain clotting factors, causing wounds that will not stop bleeding, and internal injuries that can become life-threatening. From what would in a healthy person be a minor bruise primarily affects males. Women who carry the gene on one of their two X chromosomes often show no symptoms at all. They carry the mutation silently, unknowingly, and can pass it to their children without any outward sign that anything is different about their own blood. Victoria, it appears, never knew. Neither did her doctors. Neither did the courts of Europe, who were at that very moment busily arranging marriages between Victoria's children and the royalty of Germany, Russia, Spain, and beyond. Let's trace the path. Victoria had 9 children. She was a carrier, which means roughly half of her daughters would also carry the gene, and half of her sons might have the disease itself. Her son, Leopold, Duke of Albany, had haemophilia. He died at the age of 30 from blood loss after a fall. Her daughter Alice carried the gene. Her daughter Beatrice carried the gene. Alice married into the German royal family. Her daughter Alexandra, Victoria's granddaughter, carried the gene and eventually married Tsar Nicholas II of Russia. In 1904, Alexandra gave birth to her first and only son, Alexei, the heir to the Russian throne. Alexei's umbilical cord was still bleeding abnormally at several days old. Within months, his doctors had confirmed what everyone feared. The Tsarevich had haemophilia. I want to pause here for a moment because I think it is easy to read this as a clinical case study and miss the human dimension entirely. Imagine being Alexandra. You are the empress of the largest country on earth by landmass. You're the granddaughter of the woman who ruled the greatest empire of the era. You have waited years for a male heir. Your first 4 children were all daughters, and the pressure on you had been enormous and unrelenting. And finally you have a son. And then you watch him bruise. You watch a small tumble in the nursery become a screaming hours-long crisis as blood pools beneath the skin of his knee and will not stop. You watch him cry in pain from internal bleeding that no doctor in the world in 1904 knows how to treat. You are desperate. And into that desperation walks a man named Grigory Rasputin, a Siberian peasant mystic, and depending on who you ask, fraud healer, manipulator, visionary, or some bewildering combination of all 4. Whatever Rasputin was, he appeared to calm Alexei's episodes. No one has ever been able to fully explain why. Some researchers have suggested that Rasputin, through hypnosis, or through instructions to reduce the administration of aspirin, which was commonly used for pain at the time, and which catastrophically thins the blood, may have genuinely reduced Alexei's symptoms. Others believe it was simply the power of faith of a mother's desperate hope made manifest through a man who understood what desperate people needed to feel. But here is what is not in dispute. Rasputin's presence in the Romanov household became a political liability of extraordinary proportions. The public saw an uneducated Siberian peasant with a deeply unsavory personal reputation, apparently holding sway over the Empress of Russia. Rumors spread about the relationship, about the reasons for his access, about who was really making decisions inside the palace walls. The legitimacy of the Romanov dynasty, already under tremendous pressure from a country in the early stages of revolutionary unrest, eroded further. When Rasputin was finally murdered in December of 1916, in a scene so chaotic and improbable that it still reads like dark fiction, the damage was already done. Less than 3 months later, Tsar Nicholas II abdicated. Within 18 months, the entire Romanov family was dead. Now, I want to be careful here because I am not saying that haemophilia caused the Russian Revolution. Historians have debated the relative importance of Rasputin for over a century, and most agree that the revolution was driven by forces far larger than any single personality. But the haemophilia is not irrelevant either. It created a specific private crisis inside the most powerful family in the country. It gave a desperate mother a reason to trust a man she should not have trusted so completely. It introduced an unstable element into an already unstable system at the worst possible time, and it all traced back in the end to a single, spontaneous genetic mutation believed to have occurred in Queen Victoria or one of her parents sometime around 1818 that spread across the crowned heads of Europe through the very marriages that were intended to strengthen royal power. The thing the royals used to consolidate their dynasty's marriage within a carefully managed circle of select bloodlines became the vector by which a single genetic vulnerability crossed borders, evaded all detection, and eventually helped bring down an empire. Let me step back and offer you a perspective here that I find genuinely fascinating, because what we've been circling around in the stories of the Habsburgs and the Romanovs is something much older than any royal dynasty. The idea that blood is special, that blood carries identity, that blood passed from parent to child is the vessel of something essential that cannot be seen with the naked eye. Every royal court in European history operated on this belief. The divine right of kings was at its core a claim about blood. It was the assertion that something in the bloodline of a certain family made them uniquely qualified to hold power. That the invisible stuff flowing through their veins was different from the invisible stuff flowing through everyone else's. And here is the uncomfortable irony. They were right that blood carries invisible information. They were right that it passes from generation to generation. They were right that it can determine the fate of children not yet born. They were just completely wrong about what kind of information it carries and what that information means. Blood does not carry divine right. It does not carry greatness or worthiness or the mandate of heaven. What it carries is genetics. And genetics, as the Habsburg jaw and the Romanov haemophilia can both attest, is entirely indifferent to titles. Are you someone who has ever looked at your own medical history and wondered what invisible things might be travelling through your own bloodline? Most of us have at some point. Most of us have had a doctor ask about family history of heart disease, of certain cancers, of conditions that seem to cluster in certain families and not others, and felt that particular strange feeling of looking into the past and finding it looking back. Blood types are part of that, and Rh type, as we have been exploring, is one of the most historically loaded pieces of that inheritance. Now, there is one more dimension of the royal blood story that I have been holding back because I think it deserves its own moment, and that is the question of what the royals themselves may have understood about blood, not in the scientific sense, but in the intuitive, almost spiritual sense long before any of the science existed to explain it. Because across multiple royal traditions in European history, there is a recurring anxiety about blood mixing, not just inbreeding, but the opposite, the fear of foreign blood. The Habsburgs were famous for keeping their marriages within a tight circle, but even families who married more broadly, the British royals, the French Bourbons, the dynasties of Northern Europe maintained anxious rituals around bloodline documentation. Genealogies were among the most carefully guarded documents in any court. Accusations of illegitimacy were among the most devastating political weapons. The concept of mésalliance, a marriage below one's station, was treated not just as a social failing, but as a kind of biological contamination. I'm not suggesting those anxieties were scientifically justified. They were not. Genetic diversity is generally a strength, not a weakness, and most of the harm done by royal marriage practices came precisely from too much sameness, not too much difference. but I find it interesting that the obsession existed, that across centuries and across cultures, the people who sat at the top of the social hierarchy kept returning to this idea that blood was particular, that it carried something, that it mattered in ways they could feel but could not articulate. They were looking in the wrong direction and with the wrong tools at something real. We have travelled a long way through history tonight, from a cave painting in southern France to the Habsburg court in Madrid to the palace rooms of St. Petersburg, where a desperate empress kept her son secret and opened a door she perhaps never should have opened. And everywhere we go, blood keeps showing up as the thing at the centre of the story, not as metaphor, as reality. Now here is what I want you to hold on to as we move forward. All of the stories we have just talked about, the Habsburg collapse, the Romanov tragedy, involve blood as a source of hidden vulnerability, a thing that seems normal on the outside but carries quietly something that can change everything. That is exactly the same quality we've been exploring in arch-negative blood from the very beginning. Something that does not announce itself, something that runs invisibly through generations, something that only becomes visible in a specific kind of encounter, a specific biological moment where two incompatible things meet. And that biological moment, the one that involves a mother, a child, and the most intimate possible conflict between two sets of blood, is something we have not yet fully explored. We have touched on it, but we have not lived inside it. That changes next. Because the story of what happens when an Rh-negative mother carries an Rh-positive child, told in full with all its scientific depth and all its human weight, is one of the most quietly staggering things I've ever spent time thinking about. And it is waiting just ahead of everything in this story, and we have covered a lot of ground by now. This is the part I find the hardest to fully sit with. Not because it is frightening in the way horror movies are frightening, but because it involves something that should be by every right one of the most protected relationships in biology, a mother and her unborn child, and yet in certain quiet invisible circumstances, the mother's own body fights against that child, not from malice, not from failure, from a kind of terrible misguided loyalty to itself. This is the pregnancy paradox of Rh-negative blood, and I want to take real time with it tonight because it is not just a medical fact. It is a human story, one that has touched far more families than most people realize, and one that until very recently in the span of history ended in grief far too often. Let's walk through what actually happens, slowly, carefully, because the biology here is worth understanding not just in the abstract, but as the experience of a real body doing what it was designed to do. A woman who is Rh-negative becomes pregnant. Her partner is Rh-positive. Because the Rh-positive gene is dominant, there is a strong likelihood, not a certainty, but a meaningful probability that the baby growing inside her has inherited his Rh factor. The baby, from its very earliest cellular formation, is building red blood cells that carry the Rh protein on their surface. The mother's red blood cells carry no such protein. For most of the pregnancy, this difference is irrelevant. The baby floats in its own enclosed world, the placenta acting as a sophisticated biological barrier between two distinct circulatory systems. The mother's blood and the baby's blood do not mix under normal circumstances. They run in parallel, exchanging nutrients and oxygen through a membrane so carefully calibrated that it would be genuinely impressive if it were invented rather than evolved. For months everything proceeds. The baby grows, the mother grows with it, and then comes delivery. During labour and the process of birth, particularly during the moment when the placenta separates from the uterine wall, small quantities of the baby's blood can escape into the mother's bloodstream. Just a few cells sometimes, a tiny leak in a carefully maintained boundary. For an Rh-positive mother, those few cells are no different from her own. Her immune system notices nothing unusual and moves on. But for an Rh-negative mother, her immune system encounters something it has never seen before: red blood cells wearing a protein it does not recognize, and it does exactly what a vigilant immune system is supposed to do. It notices, it catalogues, it builds a response. Quietly over the weeks following delivery, her body manufactures antibodies specifically designed to target and neutralize the Rh protein. She is now what doctors call sensitized. Her immune system has been introduced to the enemy. It will not forget. Now here is where the story takes its most painful turn. The first pregnancy, as I just described, often ends fine. The mother's immune response develops slowly enough that by the time meaningful antibody levels build up, the baby has already been born. So the firstborn goes home healthy. The family celebrates. And 2 years later, or 3, they decide to have another child. This second baby, if also Rh positive, enters a very different environment than the first, because this time the mother is not encountering the Rh protein for the first time. She's been waiting. Her immune system has essentially a pre-built weapons cache ready to deploy the moment it detects the same foreign protein again. What immunologists call a secondary immune response is faster, stronger, and more targeted than the first. The body produces a different class of antibody, this time small agile molecules that can cross the placental barrier and enter the baby's circulation. And when they do, they begin destroying the baby's red blood cells, not all at once, not in a single catastrophic event, but steadily, cell by cell, with the patient merciless efficiency of a biological process that does not know it is destroying something it was meant to protect. The baby's body responds by pumping out more red blood cells as fast as it can, but immature ones, cells that haven't finished developing, flooding into the bloodstream in a desperate attempt to keep oxygen moving through a system that is being dismantled from the inside. This is what Louis Diamond described in 1932 when he gave the condition its name, erythroblastosis fetalis, an army of immature blood cells mobilized in a losing fight. In mild cases, the baby is born jaundiced, yellowed by the bilirubin released when red blood cells are destroyed and with moderate anemia that can be treated. In severe cases, the anemia becomes so profound that the baby's heart begins to fail trying to compensate. Fluid accumulates in the body cavities. The liver and spleen swell. In the most devastating cases, the baby does not survive to be born at all. I want to stop here and just acknowledge something. If you are an Rh-negative woman, or if someone you love is, there is a fair chance you know some version of this story personally. Maybe you received the RhoGAM injection during your own pregnancies and understood vaguely what it was for. Maybe you were never told the full story of what it was preventing. Maybe you know someone whose pregnancy in an earlier era or in a country with limited medical access ended in a way that was never fully explained to them. These things are not abstract to millions of women. They are personal. They are part of family histories that were lived through sometimes without any vocabulary to describe why they were happening. And for a very long time, centuries really, they happened in silence. The case that changed everything came in 1939. A woman in the United States had lost a baby. The delivery had been complicated, and afterward, to help her recover from blood loss, her doctors gave her a transfusion. The donor was her own husband. Same ABO blood type. Should have been completely safe. Within minutes, she reacted violently to her husband's blood chills, back pain, the unmistakable signs of a severe transfusion reaction. Her doctor, Philip Levine, was baffled. He ran every test he knew. Nothing in the established science of blood typing explained why a woman would reject a transfusion from her own husband. He sent samples to Karl Landsteiner, who was in the middle of his work on the newly identified Rh factor. And the puzzle pieces fell together. The woman was Rh negative. Her husband was Rh positive. their baby had been Rh positive. During the delivery, some of the baby's blood had entered her circulation. Her immune system had built antibodies against the Rh protein. When those same antibodies encountered her husband's Rh positive blood during the transfusion, they launched an attack. She was not rejecting her husband's blood. She was responding to her own baby's ghost preserved in antibody form, still circulating in her veins. The grief of losing the child and the biological memory of that child encoded in her blood had merged into the same moment. I've thought about that quite a bit. There is something almost unbearably poignant about it. Once the mechanism was understood, the question became, how do you stop it? The answer, when it finally arrived, came from Columbia University in New York City through the work of 3 researchers, Dr. Vincent Frieda, Dr. John Gorman, and Dr. William Pollock, working in the early 1960s. Their insight was, in retrospect, almost elegantly simple. The immune system only builds antibodies against a foreign substance if it has time to encounter and process it. What if you could get there first? What if you could introduce pre-made antibodies against the Rh protein into the mother's system immediately after delivery, before her own immune system had finished recognizing and responding to the baby's blood cells? The pre-made antibodies would intercept the Rh positive cells and destroy them before the mother's immune system had a chance to register them as a threat. No recognition, no sensitization, no memory built, and therefore no attack during the next pregnancy. They tested it. It worked. In 1968, the United States Food and Drug Administration approved the resulting drug. It was called RhoGAM. Time magazine, in its coverage that year, listed it among the top 10 medical achievements of the decade. The first person to receive the officially approved treatment was a 29-year-old woman from Teaneck, New Jersey, named Marianne Cummins. She was pregnant with her 3rd child. Her previous 2 pregnancies had been Rh-positive babies, and she had not yet become sensitized. She was, in medical terms, still at risk, but not yet compromised. She had read about the new treatment in a newspaper article. The drug was scheduled for release on the 1st of June. Her due date was the 27th of May, by 4 days. Marianne made her feelings known to her doctor. Her doctor made inquiries. The release date was moved up. On the first day the drug was available, not even hours after approval, Marianne Cummins received her injection. She gave birth to a healthy baby. Decades later, she stood at a celebration of the drug's 50th anniversary at Columbia University and told her story to a room full of doctors and researchers who had spent their careers building on the foundation of that single moment. A woman who nearly missed the medication by 4 days, standing in a room full of people who understood exactly what 4 days had meant. The numbers that followed that 1968 approval are difficult to absorb without stopping. Before Rho-Gumarães disease was causing significant fetal and newborn deaths every year in the United States alone. The 2-dose protocol, a shot at 28 weeks of pregnancy, and another within 72 hours of delivery, reduced the sensitization rate in at-risk pregnancies from roughly 15% to just 0.2%. Let me put that differently. A condition that was causing widespread preventable tragedy was reduced by about 99% through a single injection given at the right moment, by doctors who understood the biology well enough to interrupt it. Today, in any country with standard prenatal care, Rh disease in newborns is so rare that many obstetricians have never personally treated a severe case. That is one of the quiet triumphs of 20th century medicine. Quiet because it does not have the drama of a surgical breakthrough or the cultural visibility of a vaccine campaign, but enormous if you understand what came before it. Now here's something I find genuinely puzzling, and I say this not as a scientist, but as someone who has spent a great deal of time thinking about this specific mystery. The urchin compatibility between mother and child is, biologically speaking, a phenomenon unique in its severity to humans. Yes, other animals have blood group systems. Horses, dogs, cattle. They have their own antigens and their own incompatibility risks. Veterinary medicine deals with blood typing for exactly this reason, but the specific human scenario where an Rh-negative mother and an Rh-positive father can produce a child whose blood type triggers a maternal immune response capable of destroying the pregnancy is not widely observed as a persistent biological challenge in any other primate. Every other primate carries the Rh factor as a matter of biological fact. There are no Rh-negative chimpanzees in the wild, no Rh-negative gorilla populations, no Rh-negative rhesus monkeys. Only in humans does this division exist. Only in humans does the question of which version of a single protein a child inherits from its father become something that a mother's immune system can turn into a biological confrontation. This does not prove anything exotic about the origins of Rh-negative blood, but it does add one more layer to a question that the more you examine it, keeps refusing to stay neatly answered. Let me ask you something directly, if I may. Have you ever looked at your blood type card or your donor registration or a lab report from a routine checkup and seen those two letters and wondered what they actually mean? Because here is what they mean in a very real sense. They mean that the protein on your red blood cells either matches something ancient and shared across almost every primate on Earth, or it does not. If you are Rh positive, you carry a marker that connects you biologically to a lineage of life stretching back millions of years. If you are Rh negative, you carry the absence of that marker. And that absence, as we have been tracing together across this long journey, is not a trivial detail. It has shaped families. It has ended dynasties. It has been carried in the bones of Ice Age hunters preserved in the isolation of mountain communities and written quietly into the medical records of millions of women who were told only that they needed a shot during pregnancy, without anyone explaining what it was for or where the story behind it began. The medical chapter of this story ends in something close to triumph. But the biological mystery is not resolved by the invention of a drug. Knowing that we can prevent the harm caused by Rh incompatibility is not the same as knowing why the incompatibility exists. And that question, the evolutionary question, the one that asks why a genetic trait that could harm offspring managed to survive 40,000 years of natural selection and still runs in the blood of 1 in every 7 people alive today, that question has a much stranger, much more fascinating answer waiting for it. One that involves the way selection actually works, the way populations survive not just despite their differences, but sometimes because of them, and the way ancient blood, once you understand what preserved it, begins to look less like a mystery and more like a message. That is where we are headed next. Somewhere in the vast machinery of human evolution, there is a rule that most biologists will tell you operates with something close to ruthless consistency. Genes that reduce survival or reproduction get selected against. They become rarer generation by generation. Given enough time, they disappear. This is not a suggestion. It is not a general tendency. It is in the framework of natural selection about as close to a law as biology has. So here is the thing that has been quietly baffling researchers since the 1940s, when the implications of the Rh factor first began to fully sink in. If Rh-negative blood can under certain circumstances in certain pregnancies put an unborn child at serious biological risk, then why after tens of thousands of years is it still here? Why does 1 in every 7 people walking the earth today still carry it? Why in some specific populations does it run at rates of 30, 35, even higher %? The uncomfortable answer is we are not entirely sure. And that uncertainty, when you sit with it long enough, becomes one of the most interesting things about this entire mystery. Let me start with what seems like the obvious explanation, and then tell you exactly why it doesn't fully work. The obvious explanation is genetic drift. Genetic drift is a process that operates alongside natural selection, but is governed not by survival and reproduction, but by chance. Here is a simple way to think about it. Imagine a small population, say a few hundred people living in an isolated valley after the last ice age. By random chance, a slightly higher proportion of them happen to carry the Rh negative allele. Not because it gives them any advantage, not because it makes them stronger or healthier or better hunters, just because out of the specific set of people who happened to survive the bottleneck, a few more of them had this particular gene variant. Now imagine that population stays isolated for thousands of years, no new people coming in, few going out. Each generation, the frequencies of their gene variants can shift slightly, just through the randomness of which individuals have children and how many. Over enough time, a trait that was present in 30% of that founding group might drift upward, or downward, entirely by chance. This is a real phenomenon. It has been well documented. It is almost certainly part of the explanation for why Rh negative blood is concentrated in specific isolated populations, the glacial refugium we talked about, the mountain communities, the island populations that carried old European hunter-gatherer ancestry without much mixing. The drift explanation is coherent. It is scientifically respectable. But here is the problem with stopping there. Genetic drift works most powerfully in small isolated populations, and the Rh negative allele is not just present in one small isolated corner of the world. It persists at meaningful frequencies across the entire Western European population, not just the Basques, but the Irish, the Scottish, the Welsh, significant portions of the French, the Spanish, the English, the Scandinavians. For drift alone to explain that level of distribution across that many distinct populations over that many thousands of years requires a very specific and rather convenient set of circumstances. And here is a deeper problem. The mathematician and biologist John Burdon Sanderson Haldane, one of the architects of the modern synthesis of evolution, a man who helped mathematically formalize the theory of natural selection, noticed something in 1942, something that has been quietly unsettling population geneticists ever since. Haldane pointed out that the way selection pressure works on the Rh system is what is called frequency dependent, meaning the disadvantage or advantage of carrying the gene depends on how common it is in the surrounding population. When Rh negative blood is rare, when only a small number of people carry it, the odds of an Rh negative woman pairing with an Rh positive partner are high, and the risk of Rh incompatibility in pregnancy is therefore high. Selection pressure against Rh negative blood under those conditions should be significant. But as the frequency of Rh negative blood in a population rises toward an intermediate level, toward roughly 50%, something mathematically strange happens. The selection pressure weakens. At intermediate frequencies, there is roughly equal probability of Rh compatible pairings in both directions, and the net disadvantage of the gene effectively approaches zero. This means that once the Rh negative allele reaches a certain frequency, natural selection essentially stops pushing it out. The gene reaches what Haldane described as a kind of neutral zone, not advantageous, not particularly harmful either at population scale, just stable, hovering, persisting, waiting. That stability argument is intellectually satisfying on one level, but it still leaves a question unanswered. How did Rh negative blood get to that frequency in the first place? How did it reach the point where the selection pressure against it was low enough to allow persistence in enough people, in enough places to account for the distribution we observe today, And this is where a genuinely fascinating line of scientific investigation enters the picture, one that involves of all things a cat. Toxoplasma gondii is a parasite, a single-celled protozoan that infects an estimated 20 to 60% of the world's human population. The exact number varies widely depending on geography, climate, and cooking habits. If you have ever owned a cat, there is a reasonable chance you have encountered it, or at least been exposed to it, though in most healthy adults it causes no obvious symptoms at all. It simply lives there, in a latent state tucked into cysts in muscle and neural tissue, dormant but present. And that is the strange part. Toxoplasma's primary goal, biologically speaking, insofar as a parasite can be said to have goals, is to get itself into a cat. Cats are its definitive host, the place where it reproduces sexually. To get from a rodent back into a cat, it does something that researchers discovered quite recently, and find deeply unsettling. It alters the behavior of the infected rodent. Specifically, it reduces the rodent's fear of cat urine. In a healthy mouse, the smell of a cat is a powerful aversion signal. Run, hide, avoid at all costs. In a toxoplasma-infected mouse, that aversion is suppressed. The mouse becomes, in some studies, almost attracted to the scent, and the mouse gets eaten by the cat, and the parasite completes its cycle. The word I have seen most often used by the researchers who study this is diabolical. I think that is fair. Now, what does any of this have to do with Rh-negative blood? In a series of studies published in the early 2000s, a Czech parasitologist named Jaroslav Flieger found something genuinely unexpected. When he separated human subjects by Rh blood type and tested them for the effects of latent toxoplasma infection, a pattern emerged. In Rh-positive people infected with toxoplasma, there were measurable changes in reaction, times the kind of slowing of psychomotor performance that the parasite seems to produce across many of its human hosts. But in Rh-negative people with the same latent toxoplasma infection, no significant effect. The Rh-negative subjects appeared protected against the behavioral and neurological effects of the parasite, and crucially in Rh-negative subjects who were free of toxoplasma infection, reaction times were actually faster than in Rh-positive uninfected subjects. There was, in other words, a measurable cognitive advantage in uninfected Rh-negative individuals, an advantage that disappeared in Rh-positive people with latent infection because the parasite was doing its work on them, and the distribution of toxoplasma infection around the world, Flieger noted, correlates in interesting ways with the distribution of Rh-negative blood. In regions where toxoplasma rates are historically high, parts of Central and Eastern Europe, areas of Africa, regions with widespread contact between humans and cats, Rh-negative blood frequencies are also elevated relative to surrounding areas. In regions where toxoplasma is extremely rare, parts of East Asia, certain island populations, Rh-negative blood is also essentially absent. Now, I want to be very careful here about what I am and am not saying. These correlations are real. The research is peer-reviewed. Flieger's work has been replicated and extended by other researchers, but correlation is not cause, and the mechanism by which Rh-negative blood might confer protection against toxoplasma's neurological effects is not yet understood at the molecular level. What we have is an observation, a striking, repeated, well-documented observation, not proof of a specific evolutionary advantage, not a finalized theory. What it is, and I think this is enough, is a plausible answer to the question that has been sitting in the background of this entire journey. If being Rh-negative carried a cost in pregnancy, under what conditions could a compensating benefit have been strong enough to keep the gene present across tens of thousands of years? The answer that is beginning to take shape is perhaps the benefit was cognitive, Perhaps in environments where parasitic infection was widespread and unavoidable, the people who carried Rh-negative blood kept sharper minds for longer. Perhaps they made better decisions. Perhaps they were better hunters, better navigators, better at the complex social calculations that govern survival in small groups. Not by a dramatic margin, not in ways anyone would have consciously noticed, but by enough over enough generations in enough environments to tip a calculation that raw numbers suggested should have gone the other way. This brings me to what I think is the most underappreciated concept in all of evolutionary biology. It is called heterozygote advantage, and it has a famous analogy that I want to spend a moment on because once you understand it, you will look at Rh-negative blood and many other problematic genetic traits completely differently. In sub-Saharan Africa, there is a genetic variant in the hemoglobin gene that causes sickle cell disease when a person inherits 2 copies of it, one from each parent. Sickle cell disease is severe and in environments without modern medical care often fatal before reproductive age. By every rule of natural selection, this gene should have disappeared from African populations long ago, and yet it persists. In some areas, as much as 20% of the population carries it. Why? Because carrying one copy, just one, makes the red blood cells slightly distorted in shape. Not distorted enough to cause disease, but distorted just enough that the malaria parasite, which reproduces inside red blood cells, has a much harder time completing its cycle. People who carry one copy of the sickle cell gene and one normal copy heterozygotes are substantially more resistant to malaria than people with 2 normal copies. In an environment where malaria routinely kills children before they reach adulthood, that resistance is worth more than the cost of the occasional child born with 2 copies and no protection against either disease. The gene persists, not because it is good, not because it is neutral, but because in the right environment at the right frequency, the benefit to the heterozygous carrier outweighs the cost to the small number of homozygous sufferers. This is balancing selection, the same principle Haldane himself suggested in his 1942 paper might apply to the Rh system. And Flieger's work on toxoplasma has given that suggestion its most concrete biological candidate yet. Here is something I find genuinely moving about this line of thinking. We've been talking throughout this entire story about blood as something that seems to defy straightforward explanation, a trait that should have been eliminated but wasn't, a distribution that seems too specific to be purely random, a cluster of populations that carried it at high rates, surviving circumstances, ice ages, invasions, demographic collapse that overwhelmed their neighbours. The evolutionary answer, if it holds up, is not mystical. It is simply the logic of trade-offs playing out over a timescale that no individual human life is long enough to witness. Every gene that persists in a population at meaningful frequency is persisting for a reason. Sometimes that reason is obvious. Sometimes it takes decades of careful research to find it. And sometimes still it has not been found at all. The Rh-negative case falls somewhere in the middle of that spectrum. We have a strong candidate mechanism. We have compelling correlational data. We have a theoretical framework elegant enough that it satisfies the mathematical models. But we do not yet have the full clean molecular story. And part of what makes this particular mystery so enduringly fascinating is that absence. The gap between we have a plausible explanation and we know is in science where all the most interesting questions live. There is one more wrinkle worth acknowledging before we move on. Everything we have discussed so far about Rh-negative blood operates within the framework of established population genetics and immunology. But there is another way people have tried to explain the persistence of this trait, one that does not involve parasites or frequency-dependent selection or glacial refugia. It involves much older questions about creation myths, about ancient texts, about what certain religious traditions and certain conspiracy theories, and yes, certain quite serious researchers have made of the fact that this blood type exists at all, and that science has not yet fully explained why. That territory is murkier, more contested, frankly much more fun to explore carefully. And it begins with a word written in ancient Sumerian cuneiform carved into clay tablets roughly 5,000 years ago, a word that translates roughly as those who came down from heaven, the Anunnaki. 1976. The Cold War is running at a slow boil. Watergate has just reshuffled American trust in authority. The personal computer doesn't exist yet, and in the lobbies of New York publishing houses a manuscript is making the rounds dense, relentless, full of photographs of ancient tablets and cuneiform inscriptions, written by a man with a kind of quiet, scholarly conviction that is either deeply impressive or deeply alarming depending on what he is claiming. His name is Zecharia Sitchin. His book is called The 12th Planet. And if you have ever spent any time in the corners of the internet where Rh negative blood is discussed with real intensity, you have encountered his shadow whether you knew it or not. Sitchin was not a crank in the obvious sense. He held a degree in economic history from the University of London. He spoke Hebrew, read ancient Sumerian, and had spent decades immersed in the cuneiform tablets and cylinder seals of Mesopotamia, the physical artifacts left behind by the Sumerian civilization that flourished between the Tigris and Euphrates rivers in what is now Iraq, roughly 4,000 to 6,000 years ago. The Sumerians are by most accounts the world's first literate civilization. They invented the wheel. They developed one of the earliest systems of writing. They created the 60-minute hour and the 360-degree circle, both of which we still use today without giving the Sumerians a moment's credit. And they wrote extensively about a group of powerful beings they called in their own language the Anunna or Anunnaki. The standard scholarly translation of this term is something close to of princely seed, or the offspring of the sky god Anu, meaning essentially the divine beings in the Sumerian pantheon. Gods. Powerful, capricious, human-shaped gods who controlled weather and harvest and the fates of kings. Sitchin looked at the same tablets and arrived at a different conclusion. His translation, which has since been challenged extensively by professional Assyriologists and Sumerologists, who largely reject it as a misreading of the original text, rendered the Anunnaki as something quite different. Those who from heaven came down. Extraterrestrial beings in his reading who arrived on Earth hundreds of thousands of years ago from a planet in our solar system he called Nibiru, which follows an extremely elongated elliptical orbit that brings it near Earth only once every 3,600 years. In Sitchin's interpretation, the Anunnaki came to Earth to mine gold which they needed for a specific atmospheric purpose on their homeworld. They used primitive hominids as labour, and eventually they performed a genetic modification combining their own DNA with that of early humans to create a worker species intelligent enough to follow complex instructions, but not intelligent enough to challenge their creators. In Sitchin's telling, we are that worker species, and a small subset of humanity, a group identifiable he and his followers suggested by the unusual nature of their blood, might carry more of the original Anunnaki genetic material than the rest. I'm going to be straightforward with you here because I think you deserve that. Mainstream scholarship does not support Sitchin's translation, not marginally, not as a matter of academic preference. Scholars who have spent their careers reading Sumerian texts, people who do nothing but study this language, and these artifacts have pointed out specific verifiable errors in Sitchin's work. The word Anunnaki does not mean those who came from heaven in any standard Sumerian reading. There is no astronomical or physical evidence for Nibiru as a real planet in our solar system, with a 3,600-year orbit. The genetic complexity required to create a functional hybrid species through genetic engineering by beings who arrived on primitive Earth and set up mining operations, the biology simply doesn't work the way Sitchin described it. And yet, his book sold millions of copies. His ideas spawned an entire ecosystem of theories, follow-up authors, YouTube channels, documentaries, academic-adjacent conferences, and devoted communities that have been active and growing for 50 years. The 12th Planet, published in 1976, is still in print. When a set of ideas that mainstream scholars reject so thoroughly generates this kind of sustained cultural momentum, I think the most interesting question is not, is Sitchin right? The evidence says he isn't, at least not in the specifics, but rather what does this story do for the millions of people who find it compelling? Because it is doing something real, and dismissing it as simply mass credulity misses what is actually interesting about this phenomenon. Let me offer you a thought. One of the most persistent human needs, maybe the most persistent across every culture and every era, is the need for our existence to mean something beyond the merely biological. We are uncomfortable being an accident. We resist the idea that consciousness and language and the experience of beauty and loss and love, all of it emerged from random chemical processes on one unremarkable planet, orbiting an unremarkable star in the outer arm of one galaxy among 2 trillion. This is not a criticism of people who feel this way. This is arguably one of the defining characteristics of our species. We reach for explanation. We reach for origin. We reach for the sense that someone somewhere made us on purpose. The Anunnaki theory taps directly into that need. It says you are not an accident. You are the product of intention, alien intention, granted and morally complicated intention involving slavery and manipulation, but intention nonetheless. And for people who carry Rh negative blood and have spent time on the internet reading about its mysterious distribution, who have felt the slightly uncanny recognition of, wait, I am one of the 15%, what does that mean? The theory adds another layer. It says you might be the ones who carry more of the original design, the ones closest to the source. I understand why that lands. I genuinely do. There is something in the human heart that wants to be told its particularity matters in a cosmic sense. And there is something especially resonant about blood as the vessel for that story, because blood already carries that weight in almost every culture and tradition that has ever existed. But here is what I find more interesting than the theory itself. The Sumerian civilization. The real one, the one we can document, is genuinely extraordinary in ways that don't require alien intervention to be astonishing. They appeared, as historians often note, with something close to bewilderment, with remarkable suddenness. Around 3,500 to 4,000 years before the Common Era, in the marshlands of southern Mesopotamia, the Sumerians developed complex irrigation systems, formal legal codes, astronomical records precise enough to track Venus across decades, architectural techniques that produce ziggurat-stepped pyramid temples that are still structurally stable after 5,000 years. Their creation myth, the Enuma Elish, describes the formation of the world from primordial chaos in language so rich and structured that scholars have compared it to the opening of Genesis, and noted the striking similarities which almost certainly reflect the fact that the biblical authors knew and were influenced by earlier Sumerian traditions. The Epic of Gilgamesh, which predates Homer by over 1,000 years, contains a flood narrative so similar to Noah's that researchers have debated for over a century which came first and which influenced which. These were not simple people. These were not a civilization that needed help from outside. They were brilliant. They were organized. They were building one of the most sophisticated societies the ancient world had ever seen. And the Anunnaki in the texts they left behind were their gods. Complex, squabbling, human-like gods, beings who got jealous and fell in love and went to war over territory, just like the gods of Greece and Rome and every other ancient pantheon. Not the cold, efficient extraterrestrial geneticists of Sitchin's telling, but something more interesting in a way, an attempt by early humanity to explain the experience of power, where it comes from, who holds it, and why ordinary people seem so often to be at its mercy. Now I want to take us somewhere slightly different, because the Anunnaki is only one thread in this tapestry. The connection between unusual blood divine origin and ancient texts runs through far more cultures than just Mesopotamia. In the Hebrew Bible, Genesis chapter 6 verses 1 through 4, there is a passage that has puzzled theologians and scholars for centuries. It is short, barely 4 verses, almost casually inserted into the narrative before the story of Noah. It says in the most common translation, "The sons of God saw that the daughters of man were beautiful, and they took as their wives any they chose. And from these unions were born the Nephilim, giants in some translations, warriors, heroes, the mighty men who were of old, the men of renown, who were the sons of God, who were the Nephilim. What happened to them the text does not say. It moves on within a paragraph to Noah and the flood, as if the whole thing were a detail, a parenthetical. But it is not a parenthetical to the millions of people who have spent serious time with it, because the Nephilim appear again briefly later in the Hebrew Bible, as the reason the Israelite spies who scouted the land of Canaan returned terrified, saying they had felt like grasshoppers in the presence of the people they had seen. Something unusual is being described, something that the authors of the text seem to take for granted their audience would understand, but which we separated from them by 3,000 years, and an entirely different cultural framework can only glimpse at the edges. I am not claiming the Nephilim were Anunnaki. I am not claiming they had negative blood. What I am saying is that the pattern is there. Across independent literary traditions separated by geography and centuries, the idea of beings who mixed with humanity, who altered something fundamental about the human lineage, who left a mark that persisted through generations in ways that could be seen but not entirely explained. Whether that pattern reflects a literal historical event, a shared mythological archetype, a recurring psychological need, or something else entirely, that is a question people far more qualified than I am are still genuinely debating. And then there is the Popol Vuh, the sacred creation text of the ancient Maya, the Quiche people of Guatemala, first written down in the 1600s, from oral traditions that stretched back centuries earlier. It describes 4 attempts by the gods to create humanity. The first humans were made of mud. They fell apart. The second were made of wood. They had no souls. The third and fourth attempts, the ones that finally worked, produced human beings of a very specific kind. The gods in one reading of the text fashion these final humans from yellow and white corn. But here is the detail that makes certain researchers pause. In the Popol Vuh, the creators worry that their final creation is too perfect, too intelligent, too aware. They can see too clearly, understand too much, perceive the full range of the god's own power and knowledge. So the gods deliberately limit the human's vision and memory. They cloud their perception so that they cannot see as far as the horizon cannot remember, as much, cannot fully understand what they are. I find that image haunting, not because I think it is literally true, but because it speaks to something real in the human experience, the sense that we are capable of more than we can access, that we carry something within us that has been in some way sealed off. Every culture that has ever grappled seriously with what it means to be human has arrived sooner or later at a version of that question. So where does all of this leave us? Honestly, in the most interesting place a mystery can leave you. The Anunnaki theory taken literally fails on the evidence. the Nephilim as historical record is beyond our current ability to verify or disprove. The Popol Vuh is myth. Profound, beautiful myth, but not genetic documentation. What none of these dismissals changes is the underlying question. Why does Rh-negative blood exist? Why does it persist? Why does it cluster in specific ancient isolated populations? Why does the body's own immune system treat it as foreign? And why does the immune system of an Rh-negative mother treat certain aspects of her own child the same way? These remain open scientific questions, and into the space created by open scientific questions, into the gap between we have some hypotheses and we know, human imagination has always poured its most powerful and enduring stories. The conspiracy theories about Rh-negative blood are not interesting because they are true. They are interesting because they reveal something about what we need. We need to believe that what is unusual about us carries meaning. We need to believe that our blood, that most intimate and universal of substances that river running through every person who has ever lived, connects us to something larger than the individual life. And in that need, I think there is actually something worth honouring. Even if the ancient astronaut theory gets the story wrong, the impulse behind it to look at the mystery in our own veins and refuse to accept that it doesn't mean anything, that impulse is not something I am willing to dismiss. But there is still one more dimension to this story that we have not yet explored. Because the theories we have just talked about, however we evaluate them all, point in a particular direction. They say that Rh-negative blood is ancient, that it comes from somewhere specific, that it follows a path across human history that can be mapped, and as it turns out, the actual scientific evidence for where Rh-negative blood travels across the world and through human migration patterns is in its own way just as strange as anything Sitchin imagined. The map is real, it is documented, and when you follow it from the Atlas Mountains of North Africa to the Canary Islands to the Scottish Highlands to the mountain communities of Sardinia, it traces a route that tells a story of ancient human movement that researchers are still right now trying to fully decode. That map is what comes next. Pull out an atlas, not a political map with its coloured country borders and capital cities. I mean the kind of map that strips all of that away, the flags and the names, the borders drawn by wars and treaties, and the ambitions of kings, and just shows you the land, mountains, rivers, coastlines, desert, the Atlantic pressing against the western edge of everything. Now imagine overlaying onto that map a single data point, the frequency of Rh-negative blood population by population region, by region drawn as a kind of heat signature across the human landscape. Where would it glow brightest? Not in the places you might expect, not in the great population centres, not in the cities where most of history's documented events have occurred. It glows brightest in the margins, in the corners and edges and isolated pockets of the world, in the places where ancient populations pressed up against a coastline with nowhere further to go, or climbed into mountains that the rest of history kept flowing around. And when you follow that heat signature from its brightest points outward, tracing the routes that connect them, what you find is not a random scatter, but something that looks, once you know what to look for, remarkably like a migration. A very old one. Let's start in North Africa, specifically in the Atlas Mountains of Morocco. The Atlas Range runs roughly 2,500 kilometers from the Atlantic coast of Morocco in the west, through Algeria, all the way to Tunisia in the east. Its peaks reach over 4,000 meters, in some places high enough to hold snow while the Sahara bakes below. In the valleys and high plateaus of this range, the Berber people, or as many of them prefer the Amazigh meaning free people, have lived for longer than recorded history can account for. They predate the Arab conquest of North Africa by millennia. They predate the Roman presence in North Africa. They predate in fact most of the political history that layered itself over this landscape. and in certain isolated Atlas Mountain communities, Rh-negative blood runs at rates that researchers have recorded at approximately 40%. 40%. That is higher than any group in Spain outside the specific Basque valleys. It is higher than any population in Ireland. It is higher by a wide margin than the average for Western Europe as a whole. A recent genomic study published in early 2026 modelled the Rh-negative allele frequencies across what researchers call the Ibero-Berber Refuge, that geographic arc connecting the Western Pyrenees with North Africa, and found that both populations show the highest global concentrations of the Rh-negative deletion, with rates of 15 to 20% in Berber populations, compared to a global average close to 15% for Caucasian populations broadly. The same study noted that ancient genomic evidence links both Basque and Berber elevated Rh-negative frequencies to Western hunter-gatherer ancestry, the same deep lineage we traced earlier in our journey to the Ice Age refugia of southwestern Europe. In other words, the Atlas Mountains and the Pyrenees sit at the two ends of a genetic corridor that stretches across the Western Mediterranean, and the blood running through the oldest isolated populations at both ends tells a remarkably consistent story. Now, there is a question here that I find genuinely irresistible. How did elevated Rh-negative blood get from the Iberian Peninsula and southwestern Europe to the Atlas Mountains of North Africa? Or, and this is the more interesting possibility that some researchers have raised, did it run in both directions, or perhaps originate somewhere else entirely and spread along routes we're only beginning to reconstruct? The most documented human movement between these two regions in ancient history runs via the sea, and here is where the story takes us somewhere I did not expect when I first started looking into this. Off the coast of Morocco, about 100 kilometers off the coast out in the open Atlantic, lie the Canary Islands. 7 main islands, volcanic, ancient, isolated enough that when European explorers arrived in the 14th and 15th centuries, the people living there had apparently lost the ability to navigate the sea that surrounded them. They had no boats capable of crossing open water, no knowledge of maritime navigation. They had in a very real sense been sealed inside their own archipelago by the ocean. And yet, they were there. Their name, the Guanches, is one of the most evocative in all of pre-colonial history. Tall, fair-skinned, by many accounts practicing mummification of their dead, speaking a language related to Berber, building structures that echoed in distant ways traditions found in North Africa and the Western Mediterranean. In 2017, researchers at Liverpool John Moores University published a landmark ancient DNA study in the journal Current Biology, analysing autosomal genomes from Guanche remains from Gran Canaria and Tenerife. The verdict was unambiguous. The Guanches were genetically most similar to modern North African Berbers. 3 of the males analysed carried the Y chromosome haplogroup, designated EM183, a lineage that is, in the words of the researchers, ubiquitous across modern North African populations and particularly common in Berber-speaking populations. The Guanches were not mysterious arrivals from the sea in the mythological sense. They were, as best the evidence can determine, Berber-descended people who made it to the Canary Islands somewhere around the first millennium before the Common Era then lost their capacity to return, stranded by the same ocean that had carried them there. And between their elevated, Rh-negative blood, their Berber genetic affinity, and the broader North African pattern they fit into the map. Now follow the trail north, past Morocco up along the Atlantic-facing coast of the Iberian Peninsula, past the Basque country, with its well-documented blood type anomaly, and continuing northward along the Atlantic face of Europe. Western Ireland, the Scottish Highlands, the Hebrides and the Orkney Islands, the western coastal fringes of Norway. Wherever the old fishing populations of Atlantic Europe have been tested, Rh negative blood runs at rates between 16 and 25% elevated, compared to the continental European interior elevated compared to populations further east. The pattern is not random. It follows the Atlantic coast as if the blood itself was pushed there by the same forces that pushed the populations carrying it ice to the North Sea to the west, later arriving peoples filling in from the east. Luigi Cavalli Sforza, one of the 20th century's most important population geneticists, mapped these distributions and noted the same gradient to west to east decline highest at the edges, where old hunter-gatherer ancestry concentrated lower toward the continental interior, where Neolithic farmer and later Bronze Age steppe ancestry diluted the earlier signal. And here is where one of the most haunting pieces of this puzzle enters. Because northwest of Britain, in what is now the shallow waters of the North Sea, between England, the Netherlands and Denmark, there is a place that 10,000 years ago was not water at all. It was land, a vast rolling expanse of hills and marshes and river valleys and forested plains. Researchers call it Doggerland, named for the Dogger Bank, a long underwater sandbank in the North Sea, the highest point of what was once an entire region, the last piece of an inhabited landscape to slip beneath the waves. At the peak of the last ice age, sea levels globally were more than 100 meters lower than they are today. What is now the North Sea was dry. You could walk from what is now Yorkshire in northern England to what is now the Netherlands without getting your feet wet. Doggerland was according to archaeologists who have spent years mapping its submerged topography using seismic survey data originally gathered by oil companies, a landscape of extraordinary abundance. Rivers, marshland, dense forest, lakes rich with fish. Hunter-gatherers lived there. We know this because fishing trawlers for over a century have been pulling ancient artifacts out of the North Sea floor, stone tools, animal bones, the occasional fragment of human remains. In 1931, a fishing trawler called the Kalinda dragged up a barbed harpoon point made from red deer antler fashioned roughly 12,000 years ago by human hands from the seafloor off the coast of Norfolk. And then around 8,000 years ago, a submarine landslide off the coast of Norway triggered a catastrophic tsunami in the North Sea, the Strega Slide. The wave flooded the remaining low-lying land of Doggerland in what must have been for anyone living there an incomprehensible disaster. An entire inhabited world erased by water in a matter of hours. I want to ask you something here, and I want you to really consider it. If thousands of people were living in Doggerland hunter-gatherers with elevated Rh negative ancestry descendants of the same Ice Age populations we have been tracing throughout this story, and the sea flooded their land in a matter of hours, where did they go? They went to the high ground, west to what is now Britain, which was at that point still barely attached to the continent, but rapidly becoming an island as the sea rose. North toward the Scottish Highlands, and over subsequent generations some followed the coast south, down through Ireland, down the Atlantic face of the Iberian Peninsula, pushing into the margins that were still accessible. This is not speculation. Archaeological and genetic evidence for the post-Ice Age repopulation of Britain and Ireland from the western edge of continental Europe is well established. The populations that first substantially inhabited the British Isles after the glaciers retreated were closely related to, in some analyses effectively identical to the same western hunter-gatherer ancestry that carried elevated Rh negative frequencies. They came from the west, from the refugium, from the places where the old blood had been preserved, and they brought it with them. Here is what I find genuinely moving about following this map. We started this journey in a laboratory looking at a glass slide, a blood sample that didn't react the way it was supposed to. And now, 12,000 years and several continents later, we are tracing a line of genetic inheritance that connects an Ice Age hunter pressing a barbed harpoon through a salmon in a North Sea river valley to a Berber farmer tending goats in the Atlas Mountains, to a mummified guanche on a volcanic island in the Atlantic, to a Highland Scot and a northwest Irish fisherman and a Basque shepherd in the Pyrenees. None of these people knew each other. Most of them lived thousands of years apart. None of them could have drawn the map. But the blood did. The blood traced the route silently through tens of thousands of years of human movement, preserving a genetic signal in the most isolated and marginal places, the mountains, the islands, the Atlantic edges, precisely because those were the places where mixing was least complete, and the oldest layers of human ancestry survived intact. Now, I want to be honest about the limits of this picture. The map is real. The data are real. The west to east gradient in Rh negative frequency is a documented peer-reviewed finding that has been replicated across multiple independent studies using different methodologies. But the exact routes of migration, the specific dates, the relative contributions of different ancient populations, all of that is still being actively researched. Ancient genomics is a young science. New studies appear every year that refine and sometimes revise previous conclusions. What I can tell you with confidence is the broad shape of the story, say of Rh negative blood, is concentrated in populations that retain the highest proportions of ancient Western European hunter-gatherer ancestry. Those populations were concentrated in geographic refugia. The distribution we see today reflects tens of thousands of years of human movement, isolation, mixing and survival. That is the map the blood drew. But I want to end this part of the story with something that has stayed with me for a long time. In 1931, when the Calinder's nets pulled that ancient harpoon point up from the floor of the North Sea, the fishermen who found it didn't know what they were looking at. To them it was just an odd piece of carved antler, something strange that the sea had given up from wherever the sea keeps old things. The archaeologist who later analysed it dated it to approximately 12,000 years ago. Someone made that harpoon. Someone held it in their hands. Somebody shaped it carefully over hours, probably in the way that someone does when they need a thing to work, really work, because if it doesn't, they don't eat. They lived in a world that no longer exists. That world is now 30 meters underwater, and yet something of them persists. Not the harpoon that was just deer antler, but the blood. The blood that was in their hands as they work the antler. The blood in their children's children's following the coast, as the water rose carrying an ancient deletion in a gene, carrying it to Ireland and Scotland and the Pyrenees and the Atlas Mountains and the islands in the Atlantic, carrying it all the way to the present day, to people who don't know the name Doggerland and never will unless they go looking. The map ends at the edges of the ocean, but the story doesn't end there. Because there is still one more dimension to our negative blood that we haven't explored. Not where it came from, not why it persists, not what ancient texts say about it, but what it might mean right now today for the people who carry it. What researchers are beginning to find about the specific physical and psychological profile that seems to cluster around this blood type, and whether the oldest blood in Europe, traced now across continents and millennia, might still be quietly doing something none of us have had a name for yet. That is where we go next. At some point in this journey, and I suspect for many of you it happened quite a while ago, a specific thought crept in. Maybe it arrived quietly somewhere around the third time you heard about ancient caves and isolated mountain populations and genetic signals preserved across ice ages. Or maybe it landed louder more suddenly, like a knock at a door you hadn't realised you were standing behind. The thought was, wait, what about me if I carry this blood type? What does that actually mean for who I am? Not in the ancestral sense, not the Ice Age hunter, not the Basque shepherd, not the guanche on a volcanic island with no boats to leave by, but right now, in the body I actually live in, with the personality I actually have, with the ways I've always seemed to process the world that maybe just maybe have never quite fit the average template. I want to spend real time here because this is the part of the RH negative conversation that tends to generate the most intense response and also the most confusion. Because there are two very different conversations happening simultaneously about RH negative personality, and they are constantly getting tangled up with each other in ways that serve neither truth nor the people trying to understand themselves. The first conversation is the scientific one: small, careful, qualified, slow. The second conversation is the internet one: loud, specific, flattering, extremely satisfying if you are RH negative and quietly certain that you have always been a little different from everyone else. Let me try to untangle them honestly. Start with the internet version because you have almost certainly encountered it whether you went looking or not. According to the accumulated wisdom of 1,000 blogs, forums, YouTube comments, and well-meaning but unverified personality guides, people with RH negative blood have higher than average intelligence. They're more intuitive than most people. They are deeply empathetic, sometimes uncomfortably so. They have a heightened sensitivity to electromagnetic fields. They're more likely to feel like outsiders, as if they don't quite belong in ordinary social situations. They have a compulsive need to seek truth. They are drawn to healing professions. They often have lower than normal body temperature and blood pressure. They are more likely to have green or blue eyes, reddish or auburn hair. They may experience a sense of having a specific mission in life without being able to fully articulate what it is. I will be honest, reading that list, even knowing what I know about the lack of scientific validation for most of it, I understand the appeal. Because, and this is the crucial thing, most of those traits are things that thoughtful, self-aware people of almost any blood type would recognize in themselves if given a quiet moment to reflect. The desire to seek truth, the feeling of occasional social disconnection, the sense that your inner life is richer and more complicated than what you show the world. These are not RH negative traits, they are human traits. They are what happens when a person is paying enough attention to themselves to notice these things at all. The fact that the RH negative personality list reads like a flattering portrait of exactly the kind of person who spends time researching RH negative blood, curious, independent, intellectually driven, skeptical of easy explanations is not a coincidence. It is a very good example of something psychologists call the Barnum effect, named after the showman Phineas Taylor Barnum, who is often credited with the phrase, "There's a sucker born every minute," though actually he likely never said it. The Barnum effect describes the tendency of people to accept vague, generally positive personality descriptions as uniquely accurate to themselves. It is the same reason horoscopes feel so personally precise, because they are written to feel precisely that way. Now, here is where I want to pivot, because the story does not end with, "It's all nonsense." It is not all nonsense. And the researchers who have spent serious time on the actual biology of RH negative blood have found some things that are considerably more interesting and considerably more complicated than either the internet version or the blanket dismissal. Remember Jaroslav Flieger, the Czech parasitologist we discussed when we explored the evolutionary puzzle of why RH negative blood persists. His research program has been far wider than just the toxoplasma reaction time findings. Over the course of roughly 2 decades of work, Flieger and collaborators have published a series of studies examining real measurable differences between RH positive and RH negative individuals, differences in health markers, immune response patterns, stress reactivity, and certain cognitive and behavioral characteristics. Some of these findings are quite specific. A 2020 study published in the journal Behavioral Ecology analyzed data from roughly 5,500 subjects, a meaningful sample size, not a small pilot study, and found that RH negative subjects reported significantly higher sensitivity to certain environmental factors. They were more reactive to heat and cold, more sensitive to alcohol and medication effects, more prone to certain types of fatigue. A separate study found that Rh-negative males in toxoplasma-free conditions showed measurably faster reaction times on certain cognitive tests compared to Rh-positive males under the same conditions. A 2022 paper in the journal Genes and Immunity found that Rh-negative males showed enhanced immune responses to the influenza A virus through a specific immune pathway, a real documented biological difference at the cellular level. None of this is the Internet personality list. None of this proves that Rh-negative people are more spiritual, more intuitive, or more likely to feel like they have a cosmic mission. What it does suggest is that the Rh protein, the presence or absence of it, may have downstream effects on biology that extend considerably further than just blood transfusion compatibility and pregnancy risk. The research is not settled. The mechanisms are not fully understood. Many of the findings are preliminary, but the pattern that Rh-negative biology is measurably different in ways we're only beginning to characterize appears to be real. Now here is the question I find genuinely fascinating. If there are real biological differences in immune reactivity and environmental sensitivity, in the way certain pathogens interact with the body, then over tens of thousands of years, would those biological differences have any influence on psychology, not through some mystical direct connection between blood type and personality, but through the more prosaic, more interesting pathway of lived experience? Think about it this way. If you are someone whose nervous system is measurably more reactive to environmental stimuli, who processes temperature chemical exposures, physical sensation, and yes, perhaps certain social and emotional inputs with greater intensity, then your experience of being in the world is genuinely different from someone with a less reactive nervous system. And experiences shape psychology, not genes directly, not blood type directly, but the texture of daily life accumulated across years in a body that registers the world with a slightly different calibration. Let me pull this into a real-world frame for a moment, because I want this to feel grounded rather than abstract. Have you ever met someone who describes themselves as a highly sensitive person? The term was developed in the 1990s by psychologist Elaine Aron, who identified a trait she called sensory processing sensitivity, a genuine neurologically documented difference in how deeply certain individuals process stimulation, both physical and emotional. Highly sensitive people tend to be more aware of subtleties in their environment. They tend to process experiences more deeply. They are often more easily overwhelmed by intense stimulation. They tend to feel emotions more intensely, including other people's emotions. They often describe feeling like they don't quite fit standard social expectations around how reactive is acceptable to be. Roughly 15 to 20% of the population is estimated to carry this trait to a significant degree. Let me give you a moment to sit with that number. 15 to 20%. That is strikingly close to the global frequency of Rh-negative blood. I am not claiming these two things are the same. I am not claiming sensory processing sensitivity is caused by Rh-negative blood type. What I'm saying is that the convergence of these numbers and the convergence of how people with both traits often describe their experience of being in the world is the kind of coincidence that probably deserves serious investigation rather than immediate dismissal. No research I know of has directly examined the overlap between Rh, negative blood type, and sensory processing sensitivity as a trait. That study has not been done, which is in itself a kind of statement about where scientific curiosity has and hasn't been directed. I want to take a small but important detour here because we've been talking about the Rh factor as if personality and blood type connections are uniquely strange. They are not. For context, in Japan and South Korea, the idea that ABO blood type shapes personality is not fringe speculation. It is cultural bedrock. The system is called Ketsueki Gata, blood type personality theory, and it runs deep. Job applications in Japan have been known to ask for blood type. Dating profiles routinely include it. Parents worry about their children's compatibility with siblings and classmates based on it. Type A, organized, careful, sensitive. Type B, creative, selfish, passionate. Type O, confident, insensitive, competitive. Type AB, rational, indecisive, genius. Scientists have been studying this system for decades, and the consistent finding is that the population level ABO blood type does not reliably predict personality. Large-scale studies involving tens of thousands of Japanese subjects have found no meaningful correlation. What it predicts with remarkable accuracy is what personality traits people think they should have based on knowing their blood type, the self-fulfilling dimension of any personality system powerful enough to become culturally embedded. The blood type personality theory in Japan is in the consensus of researchers a social phenomenon rather than a biological one. I mention this because it is a useful baseline. When we ask about Rh-negative personality traits, we need to hold that Japanese ABO experience in mind as a cautionary parallel, and we need to apply the same critical rigor. The difference, and this is where it gets genuinely interesting, is that the Rh factor has actual documented biological effects in ways that ABO type largely does not at the psychological level. The presence or absence of a specific protein on the surface of every red blood cell in your body, interacting with your immune system every moment of every day, that is a different kind of variable than which of 4 blood type categories you happen to fall into. Whether those biological differences ripple upward into psychology remains an open question, an intriguing one, an unanswered one that I think deserves serious attention rather than the choice between uncritical acceptance and reflexive dismissal. Here is where I want to land because I think there is something important and genuine in the urge many Rh-negative people feel when they learn about their blood type and want to understand what it means about them. The desire to know yourself is one of the oldest human impulses there is. It is written over the entrance of the Temple of Apollo at Delphi, "Know thyself," in letters that have not changed their meaning in 2,500 years. And the particular appeal of blood as a vehicle for self-knowledge is not arbitrary. Blood is intimate in a way almost nothing else is. It is always with you. It is invisible but essential. It is both utterly personal and through inheritance through the ancient trails we've been following across ice ages and continents, also connected to something that extends far beyond the individual life. When someone discovers they carry Rh-negative blood and feels a sudden quiet recognition, a sense of, "This explains something I never had words for," I don't think the right response is to immediately hand them a fact check. I think the right response is to hold both things at once. Here is what the science actually shows, and here is the real important, not yet fully answered question underneath the thing you feel, because the question underneath it is worth asking. It is worth asking carefully, with honesty, without rushing to an answer the evidence can't yet support. There is one piece of this story we've not yet examined, the one that predates Landsteiner's laboratory and Flieger's studies and every modern research paper, the one that asks what did ancient texts, sacred texts, the ones that shaped the moral architecture of entire civilizations make of blood that was different. What is the Holy Grail really? Why does the Bible contain passages that are still debated by scholars who have dedicated their careers to understanding them? And what does the concept of sacred bloodlines, which appears independently across the Hebrew scriptures, the traditions of medieval Christianity, and the oral histories of cultures that had no contact with each other, actually refer to? That territory is older and stranger than anything we've explored so far. And it's where we go next. Somewhere in the south of France, in a small village that sits in a valley so quiet it seems to belong to another century, there is a church. It is not a famous church, not architecturally extraordinary. Nothing about the exterior suggests that inside its walls in the 1890s, a priest named Béranger Saunière discovered something, or claimed to discover something, that would ignite 200 years of conspiracy theory, fiction, documentary film, and genuine academic debate about the nature of sacred bloodlines. The village is called Rennes-le-Château, and it is one of the entry points to one of the most layered, most seductive, and ultimately most instructive cultural mysteries of the modern era. A mystery that has almost nothing to do with what people think it has to do with, and almost everything to do with what we have been talking about all along. Let us start with a word. Sangreal. It appears in medieval manuscripts, in the great cycles of Arthurian romance, in the stories of Percival and Galahad, and the quest of the Round Table Knights for the most sacred object in the Christian world, the Holy Grail. The standard understanding of this word is linguistic sangreal in Old French means holy vessel, the cup, the plate, the dish, the object associated with the Last Supper of Jesus, said to carry miraculous properties and to appear only to those pure enough to receive it. But there is another reading. In the 15th century, an English writer named John Harding first proposed, or perhaps first wrote down what others had been murmuring, that the word might be passed differently. Not sangreal, but sangreal, royal blood. The French word sang meaning blood, the word real cognate with the Spanish and Portuguese word for royal, sangreal, the blood royal. And with that linguistic pivot, something extraordinary opened. If the Holy Grail is not a cup but a bloodline, not a vessel that held something sacred, but a lineage that carried something sacred, then the entire mythology of the medieval quest narratives takes on a completely different meaning. The knights were not searching for an object. They were searching for or protecting a family. The idea was picked up, elaborated, and pushed to its most extreme expression in 1982 in a book called The Holy Blood and the Holy Grail by three British writers, Michael Bagent, Richard Lee, and Henry Lincoln. The book became a global sensation. It made the New York Times bestseller list. It sold millions of copies. It was later plundered so thoroughly by Dan Brown for his novel The Da Vinci Code that Bagent and Lee actually sued Brown for plagiarism, a lawsuit they eventually lost, though the case confirmed just how much of Brown's plot had been built directly from their work. The argument of the Holy Blood and the Holy Grail stripped to its essential claim is this: Jesus of Nazareth survived the crucifixion, or perhaps died, but left behind a pregnant wife, specifically Mary of Magdala, known as Mary Magdalene. Their descendants, carrying what the authors called the Sangreal, the blood royal, the holy bloodline, emigrated to southern France. There over generations this lineage intermarried with the Frankish nobility. Eventually it became the Merovingian dynasty, the first royal house of France, which ruled from roughly the 5th century through to the 8th century before being displaced by the Carolingians. And guarding this secret, the location and continuation of this lineage was a series of secret organizations, most prominently the Knights Templar, and behind them an even more shadowy body called the Priory of Sion. I want to tell you what the historical record actually says about the Knights Templar, because they deserve a proper introduction rather than a brief mention as props in someone else's conspiracy theory. The real story is extraordinary enough on its own. In the year 1119, after Christian armies had captured Jerusalem in the First Crusade, and thousands of European pilgrims were making their way to the holy sites, a French nobleman from the Champagne region named Hugues de Payan approached King Baldwin II of Jerusalem with a proposal. He and 8 companions wanted to form a religious brotherhood whose specific mission would be to protect those pilgrims on the dangerous roads between the coast and Jerusalem. They called themselves the poor fellow soldiers of Christ in the Temple of Solomon. The Temple of Solomon bit was important. They were given quarters on Temple Mount in Jerusalem, in the wing of the royal palace built over the site of the ancient temple. For roughly 9 years, the order reportedly consisted of only those original 9 members, which is, historians note, a very small number of men to be protecting the roads of an entire kingdom. Some researchers have suggested the Templars spent much of that first decade not patrolling roads but excavating, digging beneath the Temple Mount, looking for something. What they found, if they found anything, has never been conclusively established. But when they returned to Europe in the late 1120s and began seeking papal recognition, they came back wealthy, influentially, mysteriously wealthy. By the middle of the 12th century, the Knights Templar had become the most powerful military religious order in the Western world. They owned castles and estates across Europe. They invented what was essentially a prototype of the modern banking system, a network of credit and financial transfer that allowed pilgrims to deposit money in one country and withdraw it in another. They answered to no earthly king and paid no taxes. Only the Pope had authority over them. And then, in the year 1307, on a single Friday, the 13th of October, a date that may have contributed to the enduring unluckiness we associate with that combination, King Philip IV of France ordered the simultaneous arrest of Templar members across his kingdom. The charges were spectacular: heresy, blasphemy, idol worship, sexual deviance, denial of Christ. Most confessions were extracted under torture. The last Grand Master of the Order, Jacques de Molay, was burned at the stake in Paris in 1314. According to legend, he cursed both the Pope and the King from the flames. Both died within the year. Is any of this connected to root-negative blood? Not directly. Not provably. But here is why I am spending time with it. Because the idea of a protected bloodline, a lineage carrying something sacred that powerful institutions were trying to find, preserve or suppress, appears in the historical record with remarkable consistency. Not just in the Templar legend, not just in the Grail romances, not just in the Holy Blood and the Holy Grail, which I should be clear was ultimately exposed as having relied heavily on documents that were later proven to be forgeries, specifically documents created by a Frenchman named Pierre Plantard, who had an elaborate and entirely fabricated connection to the Merovingian bloodline. The forgery undermined the specific priory of Sion argument almost completely, but it did not undermine the deeper historical observation that the Merovingians themselves, the real dynasty, not the conspiracy theory version, made explicit and genuine claims to a special sacral nature of their kingship that went beyond normal political authority. Merovingian kings were known as the long-haired kings because they never cut their hair. Hair in many ancient traditions was considered a vessel of strength and sacred power. The bee was their royal symbol, a golden insect that appears in Merovingian tombs and was later adopted by Napoleon, who understood its ancient resonance. And the Merovingian claim to kingship was explicitly tied to bloodline in a way that few other dynasties of the period articulated so directly. Whether any of that reflects a genuine ancient heritage, or simply the political mythology that every dynasty constructs around itself, is exactly the kind of question historians argue about. Here is what I think is the most important observation to make about the entire sacred bloodline tradition. The idea that certain blood carries special properties, that lineage is not just a biological matter, but a spiritual or cosmic one, is not unique to European Christianity or medieval conspiracy theory. It is ancient, it is cross-cultural, and it reappears with enough consistency across independent traditions that it is worth asking what it is actually responding to. In the Hebrew scriptural tradition, the concept of a sacred lineage runs from Abraham through Isaac through Jacob, a line that God specifically chose, maintained and protected across generations. The Davidic line, the royal bloodline of King David, carried in the prophetic tradition a specific promise that a figure from this line would one day establish an eternal kingdom. In the Zoroastrian tradition of ancient Persia, the divine fire of Svarena, roughly translated as divine glory or royal fortune, was believed to be carried in the blood of certain dynasties and could be lost through moral failure. In Japanese imperial tradition, the emperor was believed to carry the direct descent of the sun goddess Amaterasu, making the imperial bloodline literally divine, a belief that was formally renounced only after the Second World War. In the pre-Christian traditions of Ireland and Scotland, the relationship between the land and the king was believed to operate through blood in ways that could only be maintained through specific lineages. Everywhere, across cultures with no contact, the same pattern. Something in human consciousness, it seems, insists on the idea that certain blood is different, that it carries something you cannot see, that it connects the person who carries it to something larger, older, and more essential than the individual life. Now, what does any of this have to do with Rh-negative blood specifically? The honest answer is we don't know. The conspiracy theories that connect Rh-negative blood directly to the Jesus bloodline, to the Merovingians, to the Holy Grail, they're not supported by the evidence. There is no documented connection between the Merovingian dynasty and Rh-negative blood type. There is no document showing the Knights Templar were protecting Rh-negative lineage. The Priory of Sion was a hoax, but here is what is true. The regions of Europe most associated with the Sangreal mythology, southern France, northern Spain, the area once called Septimania, are precisely the regions where Western hunter-gatherer ancestry runs highest, where Rh-negative blood clusters, where the oldest genetic layers of European ancestry have been most persistently preserved. That is not proof of anything. It may be entirely coincidental, a simple geographic overlap between where ancient populations clustered and where certain medieval mysteries happen to be set. Or it may be one more thread in a pattern that is still in the process of becoming visible. I genuinely do not know. And I think there is something valuable in sitting with that. Do you ever find yourself drawn to certain stories? Not because they are well supported by evidence, but because they feel true in a way that evidence alone cannot quite account for. I have thought about this question a lot over the course of researching this topic. There is a category of stories, the Grail Quest is one of them. The sacred bloodline mythology is another that persist for centuries, not because they describe something that happened, but because they describe something that is still happening. The human need to believe that what is essential about us is not merely biological. The human need to believe that lineage carries meaning. The human insistence that blood, the most intimate substance, the one that runs through every generation connecting us backward and forward through time, is not merely chemistry. Those needs are not irrational. They are some of the oldest and deepest things about us. And the fact that they have latched onto the specific mystery of Rh-negative blood, onto this one unusual genetic absence, this one protein that is missing from a small fraction of humanity, says something about how powerfully the mystery invites that kind of attention. We have travelled through sacred texts and fabricated documents, through warrior monks and medieval bloodline theories, through the linguistic accident that turned a holy cup into royal blood. And we're now approaching the end of this long journey, but not quite there yet, because there is one more question waiting, not about ancient history, not about sacred texts or conspiracy theories or the particular obsessions of medieval France, but about right now, about what modern genetics, the most precise, most detailed, most honest tool we have ever built for understanding where we came from, is currently discovering about the ancient blood we have been tracing across this entire story. And what it suggests about where the story is going, that is the last chapter, And it begins with a laboratory in the present day working on questions that would have seemed like pure mythology just 20 years ago. October the 3rd, 2022, Stockholm, Sweden. The Karolinska Institute, one of the world's most prestigious medical research facilities, the institution that awards the Nobel Prize in Physiology or Medicine, holds a press conference. And Secretary Thomas Perlman announces that this year's prize has been awarded to a Swedish geneticist named Svante Pääbo. The room applauds, but the real weight of the moment is not fully visible in that room, because what Pääbo did, the specific, painstaking, technically extraordinary work for which he was being honoured, was not just a scientific achievement in the conventional sense. It was in a very real way a kind of time travel. He had figured out how to read the genetic code of people who had been dead for 40,000 years, and in doing so he had changed everything we thought we knew about what we are. When Pääbo began his career in ancient DNA research in the mid-1980s, the consensus in biology was that recovering usable genetic material from ancient remains was essentially impossible. The problem was chemistry. When a person dies, the process of cellular breakdown begins almost immediately. Bacteria colonize the tissue, water seeps in and out, ultraviolet light and oxygen work steadily at the molecular structure. Over years, decades, centuries, millennia, the long chains of genetic code fragment degrade and become contaminated with the DNA of every microorganism that has passed through the remains since death. Extracting anything meaningful from that mess was like trying to reconstruct a book from a pile of confetti found at the bottom of a flooded cave after 50,000 years. In 1985, as a graduate student at Uppsala University in Sweden, Pääbo published his first attempt extracting and cloning genetic material from a 2,400-year-old Egyptian mummy. It was technically astonishing. It was also, he later admitted, substantially contaminated by modern DNA. He kept refining, kept pushing, kept building better methods for extraction, for contamination detection, for authentication, and then in 2010, using a finger bone fragment from a cave in Siberia, he sequenced the genome of what turned out to be an entirely unknown species of human, not Neanderthal, not modern Homo sapiens, something else, something that had never appeared in any fossil record, something that could only have been detected through genetics. They called them Denisovans after the Denisova cave where the fragment was found. A tiny bone, an entirely new branch of the human family tree. The implications of Pääbo's work spread outward like a shockwave, because it was not just the discovery of Denisovans that mattered. It was the revelation that came alongside it. Modern humans, you mean virtually everyone alive today outside of sub-Saharan Africa, carry Neanderthal DNA in our genomes. Between 1 and 4% of the genome of any person with European or Asian ancestry carries sequences inherited directly from Neanderthals. The two species had met. They had had children together. Those children had survived, had reproduced, had passed the Neanderthal sequences forward through generation after generation, all the way to the present day where they sit quietly in the genomes of people who've never thought once about what prehistoric encounters their ancestors had. Denisovan DNA appears in different proportions in different populations. People from Papua New Guinea and Aboriginal Australia carry up to 6% Denisovan ancestry. Tibetan people carry a specific Denisovan gene variant in a gene called EPAS1 that allows them to thrive at high altitude where most people would struggle to breathe. Not a metaphor, not a folk tradition, a measurable, functional, life-saving piece of ancient genetic inheritance identified in a modern laboratory, traced back to a species that most people have never heard of. Ancient DNA saved those lives, and we did not know it until 2010. What I want you to hold on to, and this is the thread that connects all of this back to the blood we have been following, is the principle that Pääbo's work established. Ancient ancestry is not dead. It does not stop mattering when it gets old. The DNA of people who lived 50,000 years ago is still doing things in the bodies of people alive today. It is shaping immune responses. It is influencing altitude tolerance. It is, according to research published in late 2025, potentially contributing to how long we live. That last point deserves its own moment because it is one of the most extraordinary findings to emerge from the intersection of paleogenomics and health research in recent years. In December of 2025, a team of researchers from the University of Bologna published a study in the journal Geroscience. They wanted to know whether ancient ancestry had any measurable effect on longevity in modern populations. Their setting was Italy, one of the countries with the highest concentration of centenarians in the world. As of January 2025, Italy had recorded more than 23,000 residents aged 100 or older, with more than 80% of them women. The researchers analysed genome-wide data from 333 Italian centenarians and compared it against 690 healthy control participants in their 50s from the same geographic regions. They looked at 4 ancient ancestral components that together make up the genetic heritage of modern Europeans: Western hunter-gatherer ancestry, Anatolian Neolithic farmer ancestry, Bronze Age steppe pastoralist ancestry, and ancient Iranian and Caucasus ancestry. The finding was unambiguous. Centenarians, people who reached 100 years of age, had measurably more Western hunter-gatherer ancestry than the control group. Not more Neolithic farmer ancestry, not more steppe ancestry. Specifically, the oldest layer of European genetic heritage, the lineage traced directly back to the Ice Age hunter-gatherers who sheltered in the Western refugia, who carried elevated Rh-negative allele frequencies, who painted on limestone walls and made harpoons from red deer antler. That ancestry was the one associated with exceptional longevity. Every small increase in Western hunter-gatherer ancestry raised the probability of becoming a centenarian by 38%, and the effect was especially pronounced in women. Women with higher Western hunter-gatherer ancestry were more than twice as likely to reach 100. Let that settle in. The people who carry the oldest blood in Europe, the lineage that brought Rh-negative alleles through the last glacial maximum, that sheltered in the Pyrenees and the Atlantic edges and the mountain refugia while the rest of the population's genetic landscape was being overwritten, those people on average live longer. The researchers wrote for the first time in any published study that the Villabrunner cluster, which is what scientists call the specific Western hunter-gatherer genetic signature, contributes to longevity in the Italian population. I want to be precise about what this means and what it does not mean. It does not mean Rh-negative people live longer. The study did not examine Rh blood type specifically. What it means is that the ancient population most associated with elevated Rh-negative frequencies also appears to carry genetic advantages relevant to long life advantages that are still measurable and meaningful in people alive today, tens of thousands of years after those ancient populations lived and died. Whether the Rh-negative allele itself contributes to this effect, or whether it simply travels alongside other beneficial variants in the Western hunter-gatherer genetic package, is a question that has not yet been answered. But the researchers from Bologna did note that further investigation was warranted. They specifically called for future studies to examine whether specific Western hunter-gatherer genetic variants, not just the overall ancestry proportion, but individual alleles, might be driving the longevity effect. The Rh-negative allele is among the most distinctive markers of Western hunter-gatherer ancestry in European populations. It is not unreasonable to expect that future research will look at it directly. Let me step back from the science for a moment and offer you a perspective. We began this journey in a laboratory in 1937, an elderly man with a pipette staring at a blood sample that didn't react the way it was supposed to. A quiet, baffling anomaly. And now, 80-some years later, the tools available to understand that anomaly have advanced so far that we can extract genetic code from a 40,000-year-old finger bone sequence, it completely, and identify exactly where its owner sits in the branching family tree of human evolution. We can compare the genomes of 300 centenarians against 700 controls and identify which ancient ancestral lineage correlates with living past 100. We can trace Rh negative allele frequencies through 20,000 years of population movement, from the glacial refugia of southwestern Europe to the Atlas Mountains of North Africa, from Doggerland to the Hebrides. We can look at an ancient harpoon pulled up from the floor of the North Sea and say with some confidence the person who made this belonged to a genetic tradition that still runs quietly in some fraction of the people alive on Earth today. That is an extraordinary thing. Not magical, not alien, not the product of sacred bloodlines guarded by warrior monks. Just science. Patient, cumulative, generation after generation science-building tools careful enough to hear what the blood has always been saying. Have you ever had a moment, not a dramatic one, just a quiet, ordinary one, where you felt, without being able to explain it, that you were connected to something much older than yourself? Not a memory exactly. More like a resonance. A landscape that felt more familiar than it should. An impulse towards something ancient, fire or water or open sky that seemed to come from somewhere deeper than anything you learned. I am not suggesting those moments are genetic memories in any literal sense. The science does not support that claim. But the science does tell us something that I think is worth sitting with. The choices your ancestors made to stay in the mountains, rather than descend to the plains to follow the Atlantic coastline, rather than cross the steppe to shelter in the western refugium, rather than scatter east, those choices still live in your body. Not as stories, not as culture, but as actual molecular sequences written into the code that builds you from nothing every single time a cell divides. The blood is not metaphor, the blood is memory. And yet, for all the extraordinary progress paleogenomics has made, and it has been extraordinary, genuinely one of the most rapid scientific advances of the last half-century, there are still things we do not know. We do not know exactly why our negative blood persists at the frequencies it does. The heterozygote advantage model is plausible. The toxoplasma connection is suggestive. The glacial isolation explanation is probable. The Western hunter-gatherer longevity link is intriguing. None of these individually or together constitutes a complete explanation. The mystery is smaller than it was in 1937, but it has not vanished. And science has a particular quality that I find beautiful. It does not pretend to have closed the file while the evidence remains incomplete. The researchers who published the geroscience longevity study ended their paper with a call for further investigation. The evolutionary geneticists studying the Rh locus explicitly note that the fitness effects remain uncertain. The paleogenomicists mapping ancient blood type distribution from recovered genomes continue to refine their models. The story is not finished. It is being written right now in laboratories that Landsteiner could not have imagined by scientists working with ancient bones and modern sequencers asking questions that would have seemed like pure science fiction in 1940. I want to leave you with one last thought before we reach the final chapter of this journey. There is a specific phrase that appears in the research literature when scientists discuss the persistence of unusual genetic traits in human populations. They call it ancient variation. It means a variant, a particular form of a gene, a particular deletion or alteration in the code that arose so long ago, and has been maintained at stable frequencies for such an extraordinary span of time that it cannot be explained by simple chance. It is too old to be a recent mutation, too widespread to be a local accident, too persistent to have survived without some form of selection. The Rh negative deletion, as the absence of the gene that makes the Rh protein, has been present in human populations for at least 40,000 years, possibly longer. It is ancient variation. An ancient variation in the language of population genetics is a signal that something real is happening, something that has across the full sweep of human history contributed to the survival of the people who carry it. We have not yet decoded that signal completely, but we are for the first time in history close enough to hear it. There is one more thing to say, one final thread to pull. Because after everything we have explored, the science, the history, the ancient texts, the migration routes, the personalities, the dynasties, the sacred myths, there is still a question that sits at the very centre. Not where did this blood come from? Not why does it persist? But what does it mean, not in the ancient past, not in the laboratory, but in a person's actual life to carry something? This old, this particular, this quietly extraordinary. What is it like to be one of the 7 in every 100? That is where we end. And that ending is just ahead. We have come a long way, not just in terms of time, though we have crossed tens of thousands of years together tonight, from Ice Age limestone caves to Nobel Prize announcements in Stockholm, but in terms of what we were asking. We started with a glass slide and a question that could be stated in a single sentence: why does this blood exist? And somewhere along the way, through the mountains and the migration routes and the sacred texts and the satellite-mapped seafloor of a drowned world, the question quietly changed. It stopped being about the blood in the abstract. It became about you. Or at least for many of you listening right now, that is what happened. If you are Rh negative, this story has been on some level the story of where you came from. And if you are Rh positive, it has been the story of people who walked alongside your own ancestors, people who were in some cases literally your own ancestors, just a different branch of the same tree. Either way, I want to use this last stretch of road carefully, because the ending of a story like this matters almost as much as the beginning. Let me start with something grounding, something true and specific and completely free of mysticism. 7%. That is the most commonly cited global average for Rh negative blood. In some calculations it sits at 8 or 9%. In parts of Western Europe it runs as high as 15 to 17. In the Basque country as we know it climbs past 30. In certain Atlas mountain communities toward 40. But globally, if you could somehow take a needle to every person alive on Earth and test them, roughly 7 in every 100 would come back negative. 1 in 14. In a room of 100 people, 7 would carry this. In a crowd of 1,070. In a city of 1,070,000. That is not a tiny aberration, not a statistical rounding error, not a rare disorder that affects only a handful of people in distant places. 7 in every 100 is a significant and persistent feature of the human species. It has always been there. It will almost certainly always be there, barring some catastrophic disruption to human population dynamics that no serious researcher is currently predicting. So what does it actually feel like in the lived embodied sense to be one of those 7? I have spent time with this question. And the honest answer is that it feels like very different things to different people. For some, finding out their blood type was a Rh negative was simply a medical fact noted once on a prenatal intake form, filed away, never thought about again. For others, it became the opening of a door behind which waited apparently an extraordinary amount of very intense internet content. I am being slightly playful about that, but only slightly. Because the intensity with which people engage with Rh negative identity, the communities that form around it, the way people share their blood type with something approaching pride, the way certain characteristics get claimed as Rh negative traits, that intensity is telling us something real. Not necessarily something real about the blood, but something real about the people, about all of us if we are being honest. Here is what I think the Rh negative phenomenon is really about. Human beings have an ancient, persistent, completely understandable need to understand themselves in terms of lineage. Not just family lineage, grandparents and great-grandparents, and the oral histories passed around the table at holidays, but deep lineage. The lineage that answers the question before all of recorded history, before language was written down, before any document existed, where did the particular configuration of my blood, and my body, and my way of processing the world actually come from? For most of human history, that question was unanswerable. You could be told stories. You could look at the shapes of your face and find them echoed in the people around you. You could inherit traditions whose origins were lost to time, but you could not know. Now, for the first time in human history, we can begin to actually answer it. Ancient DNA from a 40,000-year-old bone in a Siberian cave is telling us about a species of human we never knew existed. Ancient blood group frequencies reconstructed from prehistoric remains are showing us how Ice Age populations structured their genetic inheritance. The genomes of Italian centenarians are pointing back to a specific ancestral lineage, a specific population that sheltered in glacial refugia while the world was at its coldest, and carried something forward through the ice. We're living in a moment when the answer to "where did I come from?" is becoming for the first time in history a question that can be addressed with genuine empirical rigor. And Rh-negative blood sits right at the heart of that inquiry. Because it is ancient, because it is specific, because it is measurable, and because for reasons that science has not yet fully decoded, it kept showing up in the places that preserve the oldest layers of human genetic heritage. I want to tell you about a thought experiment that I keep returning to. Imagine a family living somewhere in what is now southwestern France, roughly 15,000 years ago. The ice sheets to the north are beginning to retreat slowly, generation by generation, but the climate is still harsh, unpredictable, demanding the kind of daily attention to environment that most of us wrapped in our climate-controlled lives have entirely lost. This family, a cluster of related people, maybe 15 or 20 of them, moves with the seasons. They follow the herds. They know the river crossings. They know which plants are safe in which months. They know the cave that stays dry in the autumn rains. They do not know, and will never know, that some of the blood flowing through them carries a particular absence, a deletion in a gene that will 40,000 years later cause a scientist named Landsteiner to furrow his brow over a glass slide in a New York laboratory. They do not think of themselves as having unusual blood. They think of themselves as people, as a family, as the particular, specific, irreplaceable constellation of personalities and skills and knowledge that they are. The grandmother who knows which roots to boil, the young man who can read weather in the clouds, the child who draws animals on the walls with the sureness of line that still 15,000 years later makes people weep when they see it. Now, consider that some thread of that family, some genetic inheritance, some molecular trace persists today, not metaphorically, actually, in the veins of people alive right now. People who may or may not know they carry it. People who may feel occasionally a sense of connection to something they cannot name, or who may simply go about their lives without ever thinking about it at all. Does knowing the origin story change anything? I think about that. Knowing that elevated Rh negative blood traces to ancient hunter-gatherer populations in Western European refugia, does that change anything about the person who carries it? Medically, no. The blood functions the same way. The same precautions in pregnancy apply, the same donation card, biologically perhaps slightly. The emerging research on longevity correlations on immune difference on the toxoplasma protection hypothesis, those are real phenomena that may over the next decade of research become more precisely understood. But existentially, that is the interesting question. I think there is something genuinely valuable in knowing that your particularity is not random in the sense of being meaningless, not assigned by chance in some empty, purposeless way, but shaped by the specific history of specific people, in specific places surviving specific pressures over a span of time so vast that it genuinely resists imagination. Your blood type did not appear from nowhere. It has a history, a very long one. It was carried through the ice. It was preserved in the mountain refugia while the world outside the mountains was being overwritten. It was maintained against what evolutionary theory would have predicted for tens of thousands of years through mechanisms that we are still working to fully understand. That is not nothing. Now, I want to say something about the people who are Rh positive, because there is a tendency in discussions like this one, especially the internet versions, which I have perhaps been too gentle about to frame Rh negative blood as somehow elevated, as if being one of the 7 means you are particularly special in ways the other 93 are not. I want to be clear, that is not what the evidence supports. The Rh positive population carries its own extraordinary ancestry. The Neolithic farmers who spread from Anatolia and changed the face of European genetics, their descendants are here. The Bronze Age steppe pastoralists who drove vast migrations across the continent, carrying early Indo-European languages, and fundamentally reshaping the gene pool from Ireland to India, their descendants are here. The Denisovan variants that help Tibetan people live at altitude, the Neanderthal sequences that shaped immune responses, the lineages from sub-Saharan Africa that represent the oldest layer of all human ancestry. Every person alive is a specific, unrepeatable mixture of ancient inheritance. Every person carries something that no one else carries in exactly that combination. The Rh negative story is one thread in that tapestry, an unusually well-documented thread at this particular moment in science, but not a superior one. What I genuinely love about the blood type story, all of it, not just the Rh negative piece, is what it reveals about the nature of inheritance itself. We tend to think of the past as finished, closed, something that happened to other people in other places a long time ago. But the past is not finished. It is not even past, to paraphrase a novelist who was not wrong about very much. The past is in your blood, literally, in the specific proteins on the surface of your red blood cells, in the ancient deletion that is or isn't there, in the variants that came down through Ice Age refugia, through Bronze Age migrations, through Neolithic farmers and steppe nomads and coastal hunter-gatherers following the Atlantic edge. You are carrying their inheritance, not the memory of it, the actual molecular structure. And when you have a child, if you have or have had children, you pass some portion of it forward, not all of it. A shuffled, remixed, uniquely recombined version, but something continuous, a thread unbroken from the ancient past through you into the future. We have been travelling together for a long time tonight, through caves and laboratories, through mountains and drowned lands, through sacred texts and medieval conspiracy theories, and the quiet, patient work of scientists extracting stories from ancient bones. And I find myself at the end of this journey returning to where it began, a laboratory, a scientist, 2 blood samples side by side, one that reacted to a test derived from rhesus monkey blood, one that did not. Karl Landsteiner looked at that second sample and wrote that it merited further investigation. He was 87 years into the future of the question. He died before he could fully investigate it. We are still investigating it. We have better tools than he had, better methods, a larger, richer, more precise picture of human genetic history than anyone in 1940 could have imagined. and the investigation continues. Every year, new ancient genomes are sequenced. New population studies are published. New correlations are found and tested and either confirmed or revised. The mystery is smaller than it was, but it is not resolved. And I find something genuinely comforting in that. In the fact that even with all the tools we have built, even with the Nobel Prizes and the next generation sequencers, and the reconstructed genomes of people dead for 40,000 years, some questions about what it means to be human remain genuinely, honestly open. If you are Rh negative, I hope this journey has given you something richer than the usual story. Not the alien ancestry, not the divine bloodline, not the personality traits that may or may not apply, but something more grounded and somehow more extraordinary than any of those things. The actual history, the ice and the mountains and the Atlantic edge, the people who sheltered in the refugia while the world was at its coldest, the ancient genetic signal that survived 40,000 years because somewhere in every generation someone carried it forward, that someone at some point in the chain was almost certainly your ancestor. And if you are Rh positive, I hope this story has been a reminder that your own ancestry is no less complex, no less ancient, no less layered with migrations and survivals and improbable continuities. The blood tells everyone's story. It tells it differently for each of us, but it is telling it, right now, in every cell of your body. Thank you for staying with me through this entire journey. I know it has been long. I hope it felt worth it. If something in this story moved you, if any particular thread caught and stayed, I would genuinely love to hear which one. Leave it in the comments. Not because I need the engagement in the shallow sense that word usually means, but because the comments under a video like this become their own kind of record. A record of what resonated, of what questions the story raised that it did not fully answer, of what you're now going to go look up at 1 a.m. instead of sleeping, which was, let's be honest, probably the plan all along. Know your blood type, and know a little more of what it carries. Good night.